Mishra. He was born at Varanasi. And Dr. Gyan Prakash Mishra got educated at Central Hindu Boys School, Varanasi. He did graduation from BHU, MSc and PhD from ICR, IRI, New Delhi. And he did postdoc from University of California, Riverside as Biocast Fellow. Dr. Mishra started his professional career as lecturer at JUIT Solan, then Scientist C, then Senior Scientist as ICR DGR, then ICR IIVR Varanasi. Presently, he is Principal Scientist at ICR, Indian Agriculture Research Institute, New Delhi. His major research interest includes crop improvement through conventional and molecular approaches. Dr. Mishra has developed 9 varieties in okra, lentil, and mung bean crops. He has established a prototype of national permafrost-based germplasm storage facility at Changla, Ladakh, and developed 6 genetic stocks in lentil and garden pea. Developed 4 transgenics in peanut, and also licensed 8 okra genotypes. Published more than 75 research articles, 7 review papers in various peer-reviewed journals. Also authored, edited 5 books and 35 book chapters. He is a recipient of many prestigious honors and awards like IRI Merit Medals and Best Student of the Year Award, DRDO National Science Day Award, Limca Book of Records, ISCA Pran Vora Award, Boycast Fellow, NAS Associate, NASI Membership, ISVS Dr. Harbhajan Singh Memorial Award, Dr. Rajendra Prasad Award from ICR, Dr. Ramnath Singh Award from IRI, and Dr. Harbhajan Singh Memorial Award from ISGPV. He has visited many countries like USA, Kenya, Lebanon for research and training purpose. So, sir, with this, I would like to welcome you and please share the presentation.
Thank you, Kuldeep. So, many of you already know me very well. So, let's start this topic. Is it visible to you?
Yes. Slideshow, Mr.
I'm just doing it.
Okay, fine.
So, let me just settle down with something and then. Now slides are moving also. I think things are perfectly fine. Huh? Actually, this is also changing constantly. Earlier we used to do something. So good morning, one and all. I am Gyan Prakash Mishra. I am Principal Scientist at IRI, New Delhi. So let's start with, try to understand first this topic, mapping genes and QTLs in crop, methods and applications. So before going into detail, let's have some understanding about you that how much we know right now about the difference between gene and a QTL. So can anybody tell that whether gene and QTL are same or they are different? Then we can proceed accordingly. Some of you are from genetics background also, because I find that most of the time we have some confusion regarding this. We'll be saying that genes are the part of DNA, and QTLs are also part of DNA. So how QTL and gene are different? And sometimes we write QTL and sometimes we are writing QTLs. So how to differentiate between that what is QTL and what is gene? Just tell me something so that I can go ahead with that understanding. Although I do have made a lot of slides, but I'll be going with only that much which is of some significance. Anybody? Yeah. Say something. Yeah.
This is Dr. Nedukandi, sir, from US. It has been seen that say QTLs have been different because the influence of environment is there and all. The genes that were exploited happen to be dominant genes or say very strong additive genes, which are actually in a term they are additive. But
we come to that now. But first, we wanted to just understand gene and a QTL. That's it.
Yeah. Gene
will be
very stark. When you have a distinct phenotype being contributed by a particular gene, it can be called a gene. A gene can act in additive way or say a dominant way. QTL is a summation of many genes which actually govern the traits, which are usually polygenic in nature and their effects are cumulative in nature.
Okay.
Yeah, that's the understanding, basic understanding.
Still, I didn't understand. I want to just know what is a gene and what is a QTL. So
Sir,
do you feel like QTLs are having gene or QTLs are what?
So QTLs are actually the summation of many genes put
Mhm.
together, controlling the trait.
Mhm. So
Yeah,
why
it's
there are many genes?
quantitative trait loci. So that means quantitative trait means the
Okay.
trait is not going to be governed by single or major gene or oligogenic in nature.
Mhm, mhm.
Genes which are polygenic in nature and then govern the trait, that is termed as quantitative trait, and then the loci governing that trait are called QTLs.
So which is more important? Genes are more important than QTLs. If I can go to the gene, why I am interested in finding QTLs?
Yeah, I have come across many articles wherein it says that the
Mhm.
green revolution has been realized, but the next green revolution has to come only through these QTLs or say polygenes. This is,
Mhm, mhm.
yeah,
Okay.
they are hard to breed. They are hard to
Okay.
breed.
Anybody else? Because this is what is like our understanding about genes and QTLs. So let's try to hear one more person. Somebody wanted to say something about gene and QTL. Because my understanding is that like even QTLs are also carrying gene and genes are genes. So how to differentiate between these two? Then additive and dominant, these things are like coming afterwards. Even for the any genes also there is these things are there.
the
Okay.
genomic region which are responsible for variation in quantitative traits, though those can be considered as QTL, and genes means either it may be dominant or recessive, or with other genes it can act as complementary, means with different kind of interaction. But QTL is for means continuous variation which is contributing toward that genomic segment or regions can be considered as QTLs.
Okay, so you means that if any genomic region is there, so it must be having genes, isn't it?
Sir, the genes, gene or genes, many genes, sir.
Yeah.
Hmm.
So it means that even if I am talking about QTLs, we are talking about genes only, isn't it?
Yes, sir. But they are means it is a region that pass to the means next to the progeny
Mhm.
simultaneously, or through means if there is recombination, it will go like that, sir.
Okay. So if okay, fine. So if you say now whatever you are saying, it is perfectly fitting for the genes also. It is again a genomic region. It is also passing.
Yes, sir.
Isn't it? So let's try to understand a little bit more between these genes and QTLs. And then if any genomic region is there, if that is expressing, that is the definition of a gene. Any genomic region which is present and then part of a segment of a DNA which expresses. So that particular region is called as like your gene, isn't it? If more than one gene is there, so it is called genes. So what is that thing which makes like QTLs different than the genes? I don't know from where to get this. I'm trying to get this. I don't know. अभी तो आया था. I don't know from where to get this highlighter. Do you have any control? I'm trying to get it, but I'm not able to do it. Anyways, then I'll be using mouse. Hopefully you can see the mouse itself. So let's try to first understand difference between a gene and a QTL. Okay. I'll be using pen instead. So gene is a region of like on the DNA, which is governing something. It may not be producing always a protein. Sometimes it is only rRNA, tRNA, number of RNAs are there, which is not mRNA. mRNA is going to produce tRNA. So everything is gene, whatever it is producing, isn't it? So others can keep them muted. Vilas, you can keep yourself muted. Whenever we can interact, that time you can do it. Yeah. Now, the thing is that QTL, quantitative trait loci, it will never be locus. Some books you may find, some internet you can find that it is written quantitative trait locus. That is wrong. Okay, correct word is quantitative trait loci. So as you are telling is perfectly correct that it is a region on the DNA. So suppose this is a DNA, and how to identify that region? We'll be using markers. It could be any marker, SSR marker, SNP marker, rapid marker. It could be any kind of marker. So identification of any QTL is based on Generally, flanking marker, we'll be looking at that. Suppose there is one marker 1 and there is another marker 2, and we could find that the region in between that is regulating my trait of interest. And trait is what? Quantitative trait. We'll be understanding how the difference is there, whether it is quantitative or qualitative. But right now, since it is a quantitative trait loci, so that region is governing that. Then what could be the size of region? We don't know. Sometimes it is like in MBs. MB means million base pairs. It could be in KB. It could be even more than that. It may range to any number. So we'll be looking at some example and try to understand that how these MBs are varying. So markers which is flanking a region. So if there is a suppose 2 MB, 3 MB, 5 MB region we have identified. So whatever genes are present in between, so all becomes part of your QTL. Is it clear? So suppose we have taken one example where suppose this region is of 2 MB region, okay? And then this 2 MB will be having 1, 2, 3, 4, 5 genes are present, okay? So this QTL is having how many genes? 5 genes. How we decide this genes? Because genes are like promoters are there and then terminator sequences are there, which classifies a region as a gene. Any region cannot be a gene, isn't it? Is it clear? So this way we'll be defining any region as a QTL. Based on the marker, there are different softwares will be coming to that. But before that, if it is not clear now, whatever I'm going to say, you'll be understanding something else. So any QTL can have any number of genes. There are reports where a QTL is having even up to 600 genes. Okay, more than that. That many number of genes are there. One paper where popular was studied. So it all depends. How do then this narrow down this QTL region? Any idea? I want only few genes. So what could be the way to reduce this QTL region?
So multiple cycle of crossing to induce means recombination between those QTLs.
That is recombination. And then second part is?
Second part is screening of those means recombinant population and delineating the region using markers between this region.
Yeah, correct. We need two things. One is recombination. We'll understand what we mean by recombination. And then second is the marker, which should be polymorphic. If your marker is not polymorphic, you cannot reduce your size of QTL. Okay. So QTL is a loci. It means that is the only reason, since theoretically you may find a QTL which is having only one gene. It's only theoretical possibility. Practically getting even 5 to 10 genes is very, very difficult. Most of the time our QTL regions, because it all depends on the flanking marker, and then we'll be having hundreds of genes are present. Hardly like 10 or 15 gene QTL you are going to find. Okay. So don't worry, we'll be understanding this in proper detail. Right now, your understanding should be that gene is something which is having a initiation codon and a termination codon. And then genes are of different type. It's not like that one gene is coding for only one like mRNA. Okay. There are genes which is like which may code for multiple RNAs. We'll be looking into that also. So there is a difference between a gene and a QTL. So a QTL is having, suppose let's stick to this example where there are 5 genes first. So what is our understanding? Do you feel that all the 5 is going to regulate my trait of interest? Suppose this trait is your tall and dwarf, what Mendel has taken. Okay. So what we feel that, do you feel that in this QTL region, if there are 5 genes, 1, 2, 3, 4, 5, all are going to regulate? Generally, no. Okay.
Only your slides are not moving, sir.
No, I have not moved it.
Okay.
Okay. So there are 5 genes. Once this is clear, then I'll be fast. There are 5 genes, and then what we understand that there may not be all these 5 genes are not going to regulate your trait of interest, because you have mapped a region, not exactly the gene per se. So all the genes becomes your candidate gene. There is a difference between the term gene and a candidate gene. So we'll be using all these 5 as a candidate gene. And then based on the previous knowledge which is already there by different scientists, we try to find that out of this 5, which is the correct gene or which is the more precise candidate for my trait of interest. Okay. So this is how we go with the mapping of QTL, identifying the total number of gene and then finding which are the better candidates for me. Fine. This is one. And then there are two words you will be finding in literature, QTL and QTLs. QTL itself is a plural. Okay, QTL means quantitative trait loci, never ever locus. I'm repeating it again. Then once a word is already plural, what is the need to go with QTLs? Any idea? You understand?
Both, sir.
Yeah.
QTL means quantitative trade loci and qualitative trade loci. इसलिए this is a plural.
Okay. Let's understand this another way. Like what I said, no? In case of QTL, it is means only theoretical possibilities that we are going to get one gene. Practically, we'll be having number of genes. It could be ranging in many hundreds. 600 genes are also reported. So that is the reason that we cannot call that particular locus as a locus, because it is having multiple genes. So it is decided that let's go with the QTL, loci. So this loci is a single locus, which is having multiple genes. So it is called as loci. But so what happens? Suppose you have a plant height as a trait. So one chromosome is regulating, suppose 10%, giving 10% of that. Another chromosome may be giving 40% of that trait, because this is measured as PVE, phenotypic variance explained. Because there is no way. Otherwise, if you see the Mendelian trait, it is presence absence, tall or dwarf. But here, red or white. Here, things are not like that. Plant height is there. So any region is not going to contribute 100%. If it is contributing 100% of the trait, it becomes your major gene. Since it is not contributing 100%, so how to measure it? So there are statistical ways by which we are measuring this effect of that particular region on my trait of interest. Suppose this is chromosome 1, this is chromosome 2, this is chromosome 3, and my trait is being governed by 3 different chromosomes. One locus is governing 40%, another locus is governing 10%, another locus is governing maybe some other percent, 2%, 3%. Okay. So we'll be measuring in terms of phenotypic variance explained that out of total, how much this particular region is giving for my trait of interest, which is quantitative in nature. So suppose if I got 3, so it becomes QTLs, isn't it? So what will be the case? We are always going to get like QTLs, not QTL. We'll be always finding multiple QTLs for any trait of interest. We may be finding multiple QTL for multiple trait of interest. Clear? Okay. There is some issue with this. I'll be unsharing and then resharing because it is coming to all slide. Okay, I'm just unsharing and resharing. I don't know. It is still coming. What is the way to avoid this? Yeah, now I got it. It will be coming properly. Okay. So if you see this Mendel's work, so earlier, if you see B.D. Singh's book, it is written that all the genes are present on 4 chromosomes. But right now, there is a paper in genetics in 2011. After that, I didn't find any other paper. So as of now, this Mendel's 7 traits are present on 5 chromosomes, and numbers are very fixed: 1, 2, 3, 4, and 5. And there are like, if you see in 2023, out of 7 traits which Mendel has mapped way back in 1865, means he told about that, not mapped. Still, we have cloned only 4 genes. 3 genes are not yet cloned. So finding a gene, finding a trait, and then saying that this is being governed by single gene, 2 gene, multiple gene, but to map like cloning part, it is a really difficult task. Okay. So do you know what is the genome size of P? Any idea? KB, GB, something in your mind? What is the genome size of rice?
430 MB.
How much?
430 MB.
In case of P or rice? Rice is okay. Rice is like depending on different genotypes, it is varying around 450 MB to 379, 380 MB is the size. That is very, very small genome size. In case of P, it is ranging to around towards around like around 4 point something, 4.5 GB. So that is the size. So it is a relatively bigger genome. So there are 7 traits, sheet shape, then stem length, which is also being called as like plant height, but we generally go with the stem length, cotyledon color, seed coat color, pod form, position. So if you see here, what he found is like these are all behaving in a 3:1 behavior. Although if you see that plant height, which is a tall and dwarf, which is a typical case of a quantitative trait. But since the enzyme GA3 oxidase, which is responsible for making that particular plant tall and dwarf, was the last enzyme in the pathway. So it was hit. So Mendel could get only 3:1, although this trait was typical case of your quantitative trait, but it behaved like qualitative trait. So none of the trait, whatever we know, whether it is qualitative or quantitative, it all depends like everything is running in a pathway kind of a thing. So if last enzyme or last particular gene is hit, so we sometimes see in a way like it is behaving in a 3:1. And second part is how we are taking our data. So if Mendel had measured like this tall and dwarf in all plants in centimeter, then it could have been different. But he went ahead with only that major classification. So he got 3:1 typical case. So now we can understand, since everything is going in a pathway kind of a thing. So major gene is one thing. It depends the location of the gene where this particular gene is present. Then comes the function, and then everything is running in a pathway. Okay. So earlier, the concept was of what? Genes and polygenes, that any trait is being governed by 1 gene, 2 gene, 3 gene, 4 gene, up to certain things we can measure, and then things becomes like polygene. But then there is a need to go ahead with that quantitative trait loci. So there are a few things. Just try to keep in mind like KB, MB, and GB. Many times even students do this wonder and saying that GB is gigabyte and MB is a megabyte. These are all bases, and there is a way to write like mega bases. Always M is going to be capital and B is in small. If I'm talking about megabyte, so my M and B is going to be always in capital. Okay, so this is more important for students, not for you, but still try to be like keep in mind that mega base is also called as million base pair, and then 1 million bases or base pair, both are same because we are sequencing only one fragment. But since it's always in the pairing form, so we can write either million bases or million base pair, doesn't matter. Same happens with the gigabase space, GB, MB. So if I'm saying that lentil is 4.2 GB, so it means that that much of billion base pairs are there, 4 billion base pairs are there, and rice is only 430 or like 389 MB. So only that much million base pairs are there. So let's see this figure. This is very important to understand this single gene to multiple gene, how things are moving and how we reach to the like concept like quantitative loci and not going with the polygene. Single locus, we know only 3:1 is the ratio if it is expressing in a dominant way. Suppose if trait is not expressed in a dominant way, so it comes at like 1:1, isn't it? So one is your capital A, another one is small a. So if anything is like codominance expression is there, so we can have like red is there, and then pink is there, and then your white is there. Okay. So this dominance, codominance is always going on. And in case of quantitative trait, it's never ever like a dominant kind of a thing. So every gene is contributing to some extent. It may not be like equal extent, but it will be contributing to some extent. So if 2 genes are there, 3 genes are there, and if we go on increasing, so suppose just if 1 gene is there, we'll be having 4 different combinations, but in it expressing in like 3:1 or like 4 different combinations are there. If there is a codominance, so we'll be having 1, 2, and 1 as expression, isn't it? If 2 genes are there, 4 to the power n is the formula to understand that. So total, if you count, it becomes 16, isn't it? So 16 different combinations are coming. If it is a dominant, then also we'll be getting 9, 3, 3, 1, 4 different combinations, isn't it? So if suppose things are not dominant and each locus is contributing in a different way, so we'll be having 16 different genotypic combinations. Phenotype depends on the relative contribution of each locus. Fine. This need to be very clear. And then come to the 3 locus. Again, 4 to the power 3, it becomes 64. So we'll be having 64 combination if there is only 3 locus or 3 genes are regulating my one trait. Suppose my any trait is there which is being like governed by 3 locus. You understand why this different locus is there? Because different parts are there, and then it is contributing to your trait of interest. And let's keep one example in mind, like plant height is there. So it is being contributed by different locus. And then role of environment is also there, as some of you are telling. So if we go with the many loci, so what happens? This takes a sigmoid curve, okay? And we'll be getting this kind of a trait. If you take an example of skin color, how much variations are there? You take an example of plant height or human height, all sorts of variations are there. And it is because that individual loci, multiple loci are governing. And you can understand like why multiple loci are governing, because of your different pathways are there. Different pathways are regulating a single trait. So many enzymes are there, many products are involved in giving a particular individual a height, isn't it? So if everybody is contributing, so that is how this your normal curve comes into the picture. Fine. So a single gene becomes depends on the trait of interest. If trait of interest is like quantitative trait, so this is how things move, and then we get a typical normal curve in case of quantitative traits. Fine. So polygene till the time I can count it. If I cannot count, beyond that is there, so it becomes your quantitative trait loci. A few historical understanding, then it becomes very easy to understand why there is a need for quantitative trait loci if we are going with the multiple genes. Our polygenes are already existing in that literature. So if you remember 1953 historical year, James Watson and Crick gave this double helical structure. And then tRNA was the first, which is any like segment which is sequenced. So way back in 64, many times we get confused that PCR came first and then came the sequencing. Sequencing was going very early, and PCR came way back in 1980s only, when 85, when Mullis discovered this PCR. Then Sanger discovered this Sanger sequencing, which revolutionized at that time the sequencing mechanism. Nowadays, we have much, much better sequencing chemistry. And then comes your RFLP, which is used for mapping. Here we just isolate the DNA, fragmented it using different enzyme, and then we'll look at the gel, that how the variations are there between 2 individual or multiple individual. Then came the DNA fingerprinting. Then came this PCR. PCR has done wonders. So this Mullis discovered this way back in 1985, and then a number of your marker system came into the picture. So it is the polymerase chain reaction and the marker system which has ultimately helped in the mapping of QTL. Otherwise, we are knowing that there are traits which is being governed by this. If you see this coining of term, it was done by Gelderman way back in 1975. So he made it very clear that there is a difference between only polygene and then quantitative trait loci. So he was understanding that it is this polygene may not be a correct word, although which was given by Mather, for like where we can count, as I told, 2 gene, 3 gene, 4 gene, 5 gene. So that term is polygene. But beyond that, he said that it is better to go with the term like quantitative trait loci. Okay. So any quantitative trait loci is going to have multiple genes, a number of genes. Theoretically may contain 1, theoretical only. But if you see the practically, it is going to be in large number of genes. We'll be seeing some example. You understand that why there are so many examples of genes are there. And then what is the size of gene? If you see, so largest size known is 2.3 MB. That is the human dystrophin gene is there. Otherwise, it is in KBs only. Most of the gene, whatever we are sequencing, like in case of plants or animal, they are all like in KB. And this gene is of like MB size just because of the 79 exons, those who understand exon and intron. So it's not like that original product is very, very small, but the size is very big. This is the only gene. Otherwise, if you see the gene sizes, it's only in KBs, not even like less than 1 MB, most of the cases. So if we want to like map any mRNA, we'll be sequencing a smaller fragment. We never go with the bigger fragments. Suppose if I sequence like more than 5 MB fragment, we are not going to map any like gene because your mRNAs are very, very small. Fine. So try to remember this size also, that largest gene is 2.3 MB. Otherwise, genes are very, very small in KBs only. So now things are a bit clear. like QTL is nothing but it's a chromosomal region. Okay, how we find the chromosomal region? Because just using certain markers, which contains alleles associated with the expression of any quantitative trait. Okay, for example, yield, for example, plant height, for example, human height, it could be anything. And how to find this region? Because of certain statistical software. So it is described by statistical association. Just by any normal calculation, we cannot do it. So we'll be having a lot of softwares are there, which helps in identification of these locus. And then with the variation, how to measure variation? We'll be measuring in the field itself. Suppose I have one real population, I have any kind of population. So I'll be counting like how many plants are there, what is their height, and then we'll be having marker data available. So we'll be matching these two, and then this helps in the identification of my trait of interest. Same happens with the genes also. So now things are changing. We are not only interested in quantitative trait loci, but we also try to see that out of suppose 10 genes are there, 100 genes are there, which is the actual candidate gene which is regulating my trait of interest. Okay, and that is a really big task to find the real gene in the locus. Okay, so many times if we could do it, it's okay. If you cannot do it, so we'll be going with the transfer of QTL using those flanking markers. So we are also transferring the genes which are not at all responsible for your trait of interest. But since we have mapped a region, suppose 1 MB, 2 MB, 5 MB, 10 MB, so we'll be transferring that based on that using backcross bidding method. Okay. So what is QTL mapping? It is a phenotypic, like phenotype driven approach, which identifies what? Candidate gene. Okay, there is a difference between a gene and a candidate gene. As a scientist, we will be very cautious, even as a student also, where to write candidate gene and where to write gene. Till the time we are very, very sure that out of this 5 gene, which is the actual gene, we'll be writing only candidate gene. Okay, based on the literature which is available on the internet, which is available on the NCBI, lot of papers are there. We out of suppose I have identified 100 gene. Okay, so all 100 gene in a QTL region cannot be my candidate. So we'll be siphoning based on the literature available that which gene can be QTL, means candidate gene. So suppose out of 100, I could find 10, which is having some role in defining my plant height. Okay, so only those 10 becomes candidate out of 100, which is there in my QTL region. Okay, and then from this candidate, how to identify gene? So there are 2-3 ways by which we'll be identifying the exact gene. And one is like going with the transgenic formation. You could identify 10 and go for the transgene formation of all this 10 and then see whether all this 10 or 1 or 2, which is actually giving my plant height. Okay, this is one. Another one is that CRISPR-Cas is there. So try to mutate individually these genes and then see that which gene is actually responsible for my plant height. Okay, so this is like total number of gene. This is my candidate gene, and from candidate to find actual gene is a real difficult task. So generally what happens even in crop like rice or even in Arabidopsis, where genome size is very, very small, unless until it's damn required, we go with the transfer of QTL itself. Okay, we are never interested that out of this 10 candidate gene, which is actual gene. Okay, so because that needs a lot of money, lot of effort, and then a lot of high-end techniques are required. So each case you need to have like tissue culture protocol available. Rice, it is available, you can do it. Lentil, it is not there. Even if you identify candidate gene, you cannot go for the identification of actual gene. Fine. So there are certain limitations. That is the reason that even in 2023, we don't know that out of this 7 gene, we have cloned only 4. 4 genes are cloned, 3 are not yet cloned in case of P. And Mendel discovered way back in 1865. So even to 2023, we last year like celebrated Mendel's birth centenary of 200 birth year, and still we are not knowing that all those 7 genes not yet cloned. Okay, so cloning is the ultimate, from finding a QTL, identification of candidate gene, then reaching to the final gene. Okay, so out of this candidate gene, there may be possibility of only one gene which is actually giving your trait of interest regulating. It may be possible that there are 2 genes, maybe 3 genes which is regulating. This is how we need to be very clear that what QTL is and what is my gene and what is candidate gene. Okay, QTL may harbor hundreds of genes. Out of that, 10 may be the candidate gene. how to identify candidate gene based on the existing literature, that every gene cannot be like regulating plant height now. So a lot of things are already known. So based on that, we'll be finding candidate gene, and then out of candidate gene, if we can reach to the transgenics and or by CRISPR-Cas technique, so then only we can identify the actual gene. Fine. So there are different strategies how to identify. So mapping population is always required. Okay. So either we can go ahead with the biparental mapping population or multi-parental mapping population, where we are knowing that these are the parent involved. Biparental means only parent 1 and parent 2 are there. Okay, we'll be crossing and then we'll be generating F2 population, F3 population, or it can be like real population. It could be any near isogenic line. Depending on that, whatever needs are there, we'll be making different kinds of mapping population, go for the mapping of a trait of interest. Magic is your multi-parental population, where we can go ahead with around 6 parents. So this is what we mean by mapping population. But this does not mean that we can only map QTLs in a biparental or multi-parental mapping population. We can always go ahead in any germplasm, like NVPGR is having germplasm. If we have typical variabilities there, so we'll be making association mapping panel. That itself is another class. So association mapping is a way to map any quantitative trait loci if I have a germplasm. Why? Because it is considered that any germplasm is there, ultimately they might have originated from like 2 parent or 1 parent, and then in due course of time, because of mutations and recombinations, different kinds of germplasm variability is there. So here we'll be using historical recombination in case of association mapping. And here there is a one word which is called as linkage disequilibrium based mapping. So since it is out of context as of now, so I'm not going to detail what we understand by LD. Just understand that QTLs can be mapped in any kind of population, whether it is F2 population, real population, nil population, magic population, or even in the germplasm. The strategy is going to be different. Fine. Then a few things like how to define a QTL. Okay. So QTL, one has to have some limitation, isn't it? So it was decided just by a group of scientists that if any, first understand this main effect QTL. Okay. So main effect QTL directly affects the trait expression. Okay. And just like genes are there, there also epistatic genes are there, then main genes are there. Similarly, in QTLs also a lot of interactions happens, because ultimately you can understand that QTL is nothing. It's a genomic region which is carrying the gene. So whatever applies on that gene, same is going to apply on the QTLs also. So epistatic QTLs, they interact, just like epistatic gene. So a lot of interactions are there. So there are two major groups, MQTL and epistatic QTL. So if any main effect QTLs are there, and then this main effect QTL, if it explains more than 10% of the phenotypic variance, equal to or more than that. So we'll be using this word PVE. Always you are going to see in the papers, it is written PVE, phenotypic variance explained. So plant height is there. If it is explaining around 10% or more than that, so we'll be considering them as major QTL. Okay. So all those main effect QTL, not the epistatic QTL, main effect QTL, which is governing more than 10% or equal to that, it was decided that to consider them as PBE. But if you see, actually a lot of papers are coming, and then a lot of QTL mapping and transfer is going on using backcross population. And there are cases where it is like more than 40 or so, there you can find it becomes very easy to transfer. Okay. So if suppose more than 10 is there, it's okay. There are cases we'll be looking at that. If it is explaining a very high PV, it becomes very easy. Just transfer one locus or two locus, you'll be having all trait. So it depends like what kind of trait I'm looking at and what kind of PV I'm getting and how much that particular region is regulating my trait of interest. But theoretically, if anywhere it is being asked or for understanding, anything which is more than 10 is major QTL. And I could see like last 20 years, any minor QTL, it's good to map, but transferring them and really getting any expression from them is really difficult, almost impossible. So we'll be always focusing on major QTLs only. Okay. Then comes the stable QTL, eQTL. There are different names, not going into the detail. For example, if I am having transcript, mRNA sequencing is being done, and from there, if I am identifying any QTL, we are knowing that this particular region is based on expression only, isn't it? So if that is called as expression QTL or eQTL. So a number of terms are there, need not be worried. The most important part is what is major QTL. If it is explaining more than 10 or equal to that, and practically higher is always good. There are cases where like 80%, 40%, 60%. So there it is really being used in the marker assisted breeding using backgrounds. If you see the history of mapping, I'll be a bit fast. So like Sachs, he has done way back in 1923, he told about the linkage between a seed coat color, which is a qualitative trait, and then seed size in case of Physiolus vulgaris. So things are already known. And then Thode, he told like a way to map QTLs in Drosophila based on different cytogenic stocks he was having. And similarly, first mapped QTL, if you see, using RFLP marker, this is a restriction fragment length polymorphism, a non-PCR based marker. So fruit size, pH, and soluble solid in tomato. So things are going on from long back. It's not like that, and Gelderman way back in 70s, like termed this like QTL. But then things changed really in 1980s. Okay, what happened that we know that means late 80s, 85 discovered, and then by 90s we started understanding different kinds of markers, because for QTS we need to reach to the gene of interest, to the genome. So for that we need a lot of markers, and then we need a lot of statistical packages. So these two revolutions happened during 80s, and it is still ongoing. Each year we'll be having a really good software to map, and then now we have the markers like SNPs. So we'll be covering complete chromosome, nothing is left. And then it becomes very easy to identify my gene of interest, my QTL, more precisely than ever. Okay, so again, if anybody is asking, QTL can be theoretically a single gene. It is all, means theoretically it is possible, practically it is impossible. Okay, so conceptually if asked, it could be a single gene or it could be a cluster of linked gene. This is how we differentiate between a gene and a QTL. QTL is a region. So till now, any confusion? Let's see about the mapping. What are the things required? For any mapping, same happens with the QTL mapping. Only difference is that the trait of interest. Otherwise, normal linkage mapping and QTL mapping, more or less it's same. We need a suitable mapping population. It depends what we mean by suitable, depends on my trait of interest. It should be derived from the contrasting parent. So if one parent is tall and another parent is dwarf, then only you expect that will be able to map QTLs precisely. There are a range of differences are there. So what is the best way? Whatever germplasm you have, take the extreme ones. Don't take anything which is in between. So it becomes very easy to map, because in that case, what is the case? That here, all that tall based QTLs are there. And in this case, all those QTLs are not there. Okay, so it becomes very easy. Otherwise, what is the case? There are, suppose, some common QTLs are there. If your parent 1 and parent 2 is not very much contrasting. So very much means what? Whatever you have, if you have germplasm of 1000, look at all and then try to find the extreme ones for mapping. In case of diseases, one has to be very careful, because in that case, like certain cases, we found that disease comes so like hard that we are not going to get any seed in case of one parent, especially for MIV. So in that case, we won't go with that extreme like condition. So if you are not getting seed, your population is not going to survive by screening, then how to map it? So there will be compromising. Otherwise, we'll be going with contrasting parent. Then saturated linkage map, because we are going to map genes on my chromosome. So I want my chromosome filled with the markers. That higher is the saturation, it becomes very easy to find closely linked marker. Okay, suppose one map is like this. These are that lines are my markers. Markers are present on the chromosome. So if one case is like this, another case, suppose if I have markers this way. So I don't have, like, I can map better in case of one or in case of two. Suppose one case markers are very closely spaced. Another case we don't have very closely linked markers. Markers are placed far apart. So out of these two, which one is better to map? Which population, one or two? Any idea? One, because as I was asking, like I want to go very close to my gene of interest. So it is possible if my markers are very close. Suppose if I have this combination, So I cannot go very close. Suppose my gene is here or my gene is here, but I have markers far apart. So whatever genes are there, everything is going to go with these two flanking markers. Okay. So saturated linkage map is there. So nowadays, since I told now, we have the SNPs based marker. So we go for the deep sequencing and we'll be covering everything. So we'll be having a very dense marker and we'll be reaching very close to my trait of interest. And then comes the appropriate statistical packages. As I told that each year, different kinds of packages are coming, and then we'll be using those. And don't worry, most of that our statistics fellow and then ISRI fellow, they are there to help us, and they'll be very much understanding which is the good one and take help of them to do this. Need not to understand everything on your own. So objectives are that to identify the region in case of QTL mapping affecting the trait of interest first. Suppose I have identified that this is the region which is affecting my trait of interest. This is my first objective. But as I know that multiple regions are going to be there, it's not one region because this is not a qualitative trait. So second is that what is the effect of QTL on the trait? That is my PVE. So these two things are very, very important, that what is the region and what is the PVE. Higher the PVE, better is that region to be used for QTL mapping. And minimum requirement is what? Equal to 10%. If my PVE is more than or equal to 10%, then only it comes as major QTL, or which is part of main effect QTL. Fine. I'm touching upon only very, very essential things. So what is the process? So we know that we'll be crossing diverse line and then developing different kind of mapping population. It could be double haploid, drill, F2s, not going to those detail, understanding that we'll be having some idea about the mapping population. And then once we have the mapping population and normal your association mapping panel also, okay, it could be any kind of AM panel we generally used to say. We cannot take randomly any genotype and then start mapping. There is a way to make association mapping panel from the general plan that can be used for mapping of gene, okay. Then comes your second part is your phenotyping. That is very, very essential. Genotyping is not going to be wrong. Your machine is going to give you correct data. Problem happens always with the phenotyping. It should be for QTL mapping in the replicated trials over location and year. So that's the limitation sometimes happens within case of F2 mapping population, because it cannot be replicated over location and year. But still we can map if it is very, very essential. Otherwise we try to avoid using F2s for that. For any like minor QTLs. Major QTL can be mapped using F2. Otherwise we prefer not to use F2 unless until we don't have any other option. So phenotyping replicated trial over location and year, for that we need a immortal mapping population. So just understand that RILs and double haploid, these are your immortal kind of a mapping population. So these are used, even your association mapping panel is also immortal. So these are used for multiple year and location, so that we will be getting what? Stable QTL. Otherwise, there is a chance of some error. Suppose there is a 1 year 2021 disease is not properly coming in your field, or something happened in your field, and then my data is not that correct. So second year we did it some other location or same year. So if we have 2 to 3 years data, so you are going to get a stable QTL. Okay. So which is expressing in multiple year, multiple generations, we are going to select those QTLs only for the mapping purpose. Rest other QTLs are generally discarded. Then comes your parental polymorphism, means from where you have generated this population except AM population. So we need large number of marker. Now you understand that genotyping is there. Earlier we are using generally SSR marker. Nowadays, like rarely will be going with SSR marker because your genotyping cost is so low. It is always suggested to go with SNPs as a marker, means go for the sequencing. Either you can go for GBS kind of a sequencing, genotyping by sequencing. If you have less money, or if you have a lot of money, go with the WGRS, whole genome resequencing approach, so that you will be having deep sequence data available with you, and then go for construction of framework linkage map. What we mean by framework linkage map, suppose I have lentil as a crop, so there are 7 chromosomes. First, I don't see anything like my trait of interest. I only go with SNPs and finding that chromosome 1, how many SNPs are present, and chromosome 2, chromosome 3, chromosome 4, 5, 6, and 7. Because there are 7 chromosomes just like P in case of lentil also. You can think of anything. So we'll be sequencing and then finding my marker on all these chromosomes. There is a way, like if marker to marker linkage is there, so that number of softwares are there, and then company fellow is going to give you this kind of a thing. First is this. Once we found this, and then next part is going ahead with the mapping. Okay, so first is the framework linkage map. Unless until I have my linkage map with me, I cannot map, because I need thousands and thousands of marker, maybe millions of marker if it is present. So SNPs are the best way. Otherwise, SSRs are also equally good. Like when I used to be a student, we'll be using this SSRs only for mapping. But now using SSR for QTL mapping is not suggested. So straight away go with that, and then finding the data like linkage, which part my QTL is present and how much of PV it is contributing. Okay. So this is how we do it. So what is important? Contrasting parent, stable population, multiple year, multiple location data, then a proper framework linkage map based on good markers. Okay. So if you are doing with SSR, it's okay. At least use a few hundred SSR, 600, 700, then chances are there to get it. But if you have any possibility of going with the SNPs, never use any other marker. So these are the like mapping population. Whosoever is a Samsung, please mute. So F2 is there, different kinds of marker system is there, near isogenic line, rail. Okay. So not part of this. So there are different ways like experimental crosses. We are crossing and then we are knowing the parental combination. And then another one is your association mapping population. Okay. So what we do to find the QTL, we need to marker is there and then trait is there. So for that, we'll be saying that marker trait association. We'll be finding marker trait association for the identification of QTL, because we'll be identifying a region. But here, in case of QTL, one unique thing is that is called as covariate. I will not be touching anything like much detail, because these are good for students, not for you. But still keep in mind that covariates are part in case of QTL mapping, which is not there in case of normal any kind of a major gene mapping. Like what is the age, what is the gender, what is the environmental condition, whether rains are there or not, whether it is good for the virus to be present in the field or not. Many things are there, whether some kind of drought is there or not. We use that data also while going for the QTL mapping. Okay, so if it is multiple year, multiple season data is there, we'll be taking consideration into that, so that our mapping is not going to be biased kind of a thing. Okay, it should be a precise mapping earlier. Then there are different data structure. Just understand that we'll be taking phenotype. Suppose plant height, we'll be taking plant height. Then genotype, it's the marker. It could be SSR marker, or it could be your genotype SNP based marker. Then comes the covariate, like what are the environmental factors are there, age, gender, whatever I told. And then we'll be generating genetic map and then putting all the markers together, and then we'll be clubbing all these three to get my QTL in that. Okay, so marker data, different ways are there, not going to detail like P1, P2, F1. Okay, but there are ways. Marker data is there, then marker scoring is being done, then QTL genotype, then phenotype is there, and then we'll be ultimately reaching to the physical location or genetic distance as either recombination frequency between markers, pairs as a linkage group. Okay, so we'll be finding this marker location, because if there is a parent 1 and parent 2, and then there is a crossing over, so whatever markers are there, we get that suppose F1 is there, and then we will be having RIL. So it becomes very easy to find my linkage group. Linkage group is the chromosome on which all these genes are, all these markers are present. Till the time chromosome numbers are not known, we'll be using the word LG. LG means linkage group. Okay, if chromosome is known, then we'll be calling them as chromosome. Otherwise, we'll be using the word LG, linkage group. So if there are 7 chromosomes, so total number of like if I have very saturated map, LG is going to be 7 only. Okay, it can be more or less, but if saturated map, it will be same. So there are 2 major groups for QTL mapping. We'll be touching upon just a basic understanding of that. That is single QTL mapping and multiple QTL mapping. Okay. So at a time we measure 1 QTL. So that is called like single QTL mapping. So there are 2 ways to do this. One is called single marker analysis and simple interval mapping. We'll be looking at that. What is the difference between these two? And second is that multiple QTL mapping. We'll be mapping multiple QTLs together at one time. Okay. So there are 3 major ways like composite interval mapping, CIM, multiple interval mapping, MIM, just remembering this name is more than enough for you, okay? And then Bayesian analysis. These are like 5 major ways by which we'll be measuring. Nowadays, we won't be doing any like single QTL mapping. Even when I was in MSc way back in 2000, then at that time we used to do this. Now it's completely based on multiple QTL mapping, but still certain crops are there. Nothing is known, no marker system, nothing. Though there still I could see like some single map QTL mapping is going on. So single marker analysis is what? Suppose this is my chromosome, this is the marker, and this is my QTL. Problem with this kind of study is that our marker is there. We never know whether my QTL is on the upper side or on the lower side, isn't it? Because we are looking at the marker in the phenotype. So that is the problem with your single marker analysis. Okay. So single factor ANOVA was used for mapping such kind of a trait. It is very, very preliminary. As I told you, way back, like more than 2 decades before, this was being used. And then a better version was devised, which is called as interval mapping. So what was done? We always went ahead with the interval. Okay. Try to find interval marker 1 and marker 2, and then look the QTL in between. Otherwise, what was happening in this case, there is one term called ghost QTL used to be there. So to avoid that ghost QTL, we are not knowing anything which is near to that. That also comes into the picture. I'm looking QTL this side, one more QTL is there, some other side. Okay. So that clarity is not there. We'll be transferring marker and sometimes QTLs are not going. Okay. So to avoid that, interval mapping came into the picture, and then we'll be always going with the flanking markers, so that my QTL is sitting in between that, and then this QTL is transferred. Then came your interval, like composite interval mapping, where we wanted to transfer multiple QTL rather than only one QTL, because always my trait is being governed by more than one like QTL. So it is basically interval mapping only, but then it includes multiple regression analysis, means it takes into consideration total data at a time, and it finds the QTLs on different chromosome. Okay. It could be like more than one QTL on single chromosome or one QTL on one chromosome, all the combinations. Okay. This is what is your composite interval mapping. Details are there, I have avoided because it will take like 3-4 hours to do that. This is more than enough. And then comes your multiple interval mapping. So here it is almost simple, like similar to your composite interval mapping. Only difference is that it not only looks markers just like what is happening in case of composite interval mapping, but it also takes this multiple marker interval simultaneously. What we mean by simultaneously, it looks all the chromosomes together. Here what it does, it first reads marker here, and then it goes to this part, then it comes to this part, then it comes to this part. It does mapping individually in one interval, whatever interval based on the markers we have. This interval is decided by the markers available with you. It goes like this, okay? And then it finds, okay, my QTL is there, so identifies one. Here it does this simultaneously, not only looks this QTLs on one chromosome, it is all software based. It tries to see the chromosomes like nearby also. Okay, so suppose my trait is there, suppose plant height, it is being governed at 2 locations, it's tried to find everything together. So if you see like different mechanisms are there, MIM is now being used mostly for your mapping purpose, because this is a better one and tries to avoid many things, and it tries to find QTS more precisely than any other method. Fine. So try to remember that MIM is multiple marker, uses multiple marker interval, and it tries to simultaneously fit everything, and then it is more precise than any other marker system, like mapping method. So whatever software we are using will be integrating different combinations into that. Then there is one term which is called as hotspot. So there are certain regions which is carrying number of QTLs together. So what is the advantage? Just transfer this region, and then multiple traits are being transferred together. So as I told that around 600 genes are present in a QTL hotspot in case of a popular tree. Okay. So that are used to be like transfer of that. Similarly, eQTL hotspot, eQTL means that expression, means we have identified QTL based on mRNA. So there are certain papers which says that we have identified that hotspot. Let's take 3 examples to understand that practically how it is happening. This again paper from India only, Raju Varshney group is there from ICRISAT. Now he is in Murdoch University, Australia. So they are trying to identify a region for drought tolerance in case of chickpea. Okay. So they first paper came in 2014, and then again they published another paper in 2015 using same mapping population. Okay. So what they did, they first identified 1007 markers, 20 traits, which is governing your abiotic stress tolerance or drought tolerance. And traits are like root length, density, dry root weight by total plant dry weight, root surface area, number of traits were there. I'm just mentioning a few. And then they found that there is a region which is governing most of these traits together. Okay, so they have identified a QTL hotspot region where 91 MQTLs are there for different trait. You understand? It's not for one trait. Although overall trait is your drought tolerance, but they have measured drought cannot be measured as a drought. No? So they have measured multiple traits which is governing drought. So they found that there are 91 MQTL. MQTL means what? Any idea? It is these 91 QTLs are governing more than or equal to 10% of PV for the drought. Okay, so not drought per se, but for different trait of interest. That's the reason different colors are there. Okay, blue, green, red, so many colors are there. Okay, so first year, like in 2014, they identified a locus which is 29 centimorgan. Then again, they increased with number of markers, and then they reduced this locus to 14 centimorgan in 2015. Okay. And then the cross was from ICC 4958 and ICC 1882. That was used for making this identification of this QTL. After that, since this QTL was so good, it is giving very clear-cut drought tolerance to the chickpea, and then they used for the transfer of this QTL into two parental varieties. Okay, that is DCP 92-3 and PUSA 362 from IRI. And then it took some time based on the marker assisted selection. They have the flanking marker they have used to transfer it, and then this typical QTL hotspot was transferred. Yeah, one more thing. This hotspot is actually 4 MB region. So now we understand 4 million base pairs. That was the region which is used for the transfer. So this particular region was transferred, and then based on this particular hotspot, they have released two varieties that maybe 2 years before these two varieties came released by our then Mahapatra sir was as BG. So IPC 4-14 and BGM 4005. Okay, so identification of even drought tolerance, abiotic stress tolerance QTL was there, and then it could be possible because of this QTL hotspot, means for multiple trait, whether it is root length density, root weight is there, root surface area is there, everything is being governed by this particular region. And this was a not very big region, only 4 MB region. So it was used for the transfer of trait of interest. This does not mean that only all the genes are of importance. All the genes are not candidate gene. But then if I'm transferring this, I'm getting my drought tolerance. So let's transfer even some genes, or even if they are not regulating any trait of interest. Okay, because counterpart is always there in other one. No? Okay, so comparison of QTL hotspot was there with 49 novel SNP markers were there, and then these markers are used for the transfer of trait of interest. So from identification of this way back in 2014, and then release of variety, I think 2021 or 20, something like that. No? I missed that. So ultimately this variety came into the existence using mass. Another case is there when I was there in Junagadh, that time we are trying to map a QTL which is governing Sclerotium rollsii, which causes stem rot resistance in case of groundnut. So problem with this fungus is that this is a necrotropic fungus. It kills and then it enjoys the food. So how to map that? But when we do have a very clear-cut lines were there, so we then crossed it and then went for the real population. And then this RIL was used for the mapping of gene. But since there is a complexity of genetic architecture, because this is a typical necrotropic fungus, and then for a few days, like maybe 10-15 days, resistant becomes resistant. But if you give like 30 days to feed, then this Sclerotium rollsii is going to even kill resistant line also. But susceptible goes within 10 days, less than that. So in that case, we did not find any like MQTL. We only identified 44 major epistatic QTLs. Okay, so it means that since in case of complex trait, there is only interaction going on. So PV is ranging from like 14.32% to 67.95. That much high PV is there. And once we started to like narrowing down that where these genes are present, so we found that gene is present on B4 chromosome. In case of groundnut, there are two genomes, A genome and B genome. There are two genomes are there. This is a case of allotetraploid. And then in case of B genome, there is a region of 1.8 centimorgan. But this centimorgan is the crossing frequency. So if we need to understand physical distance, we'll be always going with the MB, like how much is the actual size of this region. So what was identified that actual size is 5.2 MB. Previous case, what we have seen, 4 MB is the total region. But here we could identify 5.2 MB region, which is carrying 170 genes. Okay. So out of this 170 genes, only nearly 20 genes are of my candidate genes, okay, which includes like RRR is there, zinc finger motif, ethylene responsive factors, these are there. So ultimately, only those genes are identified, which is supposedly regulating my trait of interest, okay. So these are called as my candidate gene, okay. So there are cases where even in the real population, if trait is very, very complex, so we could not identify any major QTL, but we'll be getting only epistatic QTL, which is regulating my trait of interest to the extent of like a major gene, 67 to 68% of PVE just by one region. Then another example is there, which is case of MIMIV. MIMIV is mung bean yellow mosaic virus. In India, there are two parts for any begomovirus. One is North India, another one is Southern India. So Southern India, the isolate which is affecting is MIMV. In Northern India, it is MYMIB. Mung bean yellow, very difficult to write. Mung bean yellow mosaic India virus. Okay, there are two strains. Only little bit sequence dissimilarity is there, but that makes them altogether different. A line which may be very, very resistant here in Northern India is going to be very susceptible in Southern India. How these variations are happening, no one knows. But these are the this is how these strains exist. So they mapped this mung bean yellow mosaic virus using interspecific cross of Vigna radiata and Vigna umbellata. Okay. They identified one major QTL on chromosome number 4, and then that region is 1.28 MB region, okay, on chromosome number 4, and this is how they write MIMB4-1. So in that 1.28 MB region, 83 annotated genes were there, okay. So what I'm trying to say here, 5.2 MB region, 170 genes are there. So this MB region crossing frequency altogether different. 1.8 centimorgan in case of P, like groundnut, it is differing depending on cross to cross. It need not be always 5.2 MB. Huh? It always varies. So here also, like it's 1.28 MB, and then they find that total 83 annotated genes were there. Okay, this is the location where genes are mentioned, Vigna radiata, and then these are the numbers. Okay, out of that, they could identify 18 as a candidate gene. Okay. So we are also working on here MIMIV. I also tried to see all on chromosome 4, but I could find that in our case, our mapping population, till now, I didn't find any candidate gene on chromosome number 2, like 4. We will be getting in some other chromosome. Okay. So soon we'll be publishing that in case of MIMIV, where that major genes are located on which chromosome and how they are regulating. There is another issue with this paper is that actually they use two things, Vigna radiata and umbellata, and they are mostly comparing the sequence of Vigna radiata. But anyways, they have found like on chromosome number 4, 83 annotated gene, but how many candidate gene? 18. So now I hope you'll be more clear with the total genes which is present in my QTL region, because QTL is now defined by my flanking marker. So flanking marker says this is my QTL. I don't have any other marker in between. So I cannot use anything else. So if I want to transfer this particular region, so I'll be using my flanking marker M1 and M2, which has decided my QTL. And this is going to carry, in this case, 83 genes. Although my candidate genes are 18. Doesn't matter, no? It doesn't matter. Even if I am transferring 83 genes, at least I'll be transferring my trait of interest. This is how QTL and a normal genes are differing. Then there is another term, I'll be just finishing, QTL-seq, or this is also called as BSF or QTL mapping. Okay. So in this case, what we do, we'll be taking 5% extreme phenotype. So bulk segment analysis is an analysis in which we'll be mapping genes. We'll be mapping genes based on bulking of a DNA. Two extreme DNAs are bulk, and then becomes very easy to map. So same was being applied for QTL-seq, which is also called as bulk segment analysis of QTL mapping. Rather than going with 200, 300 plants, we'll be going with only 3 samples: bulk 1, bulk 2, and then any one parent. Okay, it could be any parent, parent 1 and parent 2. And then reference genome is being used for mapping. Just this much is more than enough to understand. And then in case of QTL-seq or BSA, which is used for the QTL mapping, in this case, what we do, will be taking like only major kind of a QTL to be mapped. Any minor QTL is generally not being mapped using this strategy. So if I'm in hurry, I want to map my major QTL. So even F2 population is considered best for this particular kind of mapping. You can use any population, but if you really want to use any major QTL using F2 population, the best strategy is QTL-seq, because it identifies your trait of interest easily, like very easily can identify. Fine. So this is how, like, so many things are there. Can I have 5-10 minutes more, or
Yes, sir, sure, you can have, sir.
Okay. So let's have a little bit more clarity, and then we'll be interacting for some time. Okay. So I'm not going more detail like what is the SNP index, high and low. Basic understanding is that QTL-seq only for major gene mapping. And if you want to map in F2 something major, go ahead with QTL-seq. Even if you have real population you want to map, you'll be looking at your field that this trait is like being governed by 1 or 2 or 3 genes, which is very easy for any breeder. We can identify that things are going on like this. So rather than going with the isolation of DNA for everything and then going for the mapping of 200 plants, it's better to go ahead with the mapping using only 3 samples. It is cost effective also. It is more precise also. Okay. So even crops like lentil will be using this kind of strategy and will be precisely mapping my gene of interest, rather than going with 600 SSRs and then isolation of DNA and then unnecessary wasting a lot of money by doing such kind of a thing. But only thing is that minor QTLs you cannot map using such strategy. There you need to go with either WGRS. WGRS is nothing but it is called as whole genome resequencing. Okay. And another one is your GBS, genotyping by sequencing. There are different ways. So it all depends on the depth of the data, how much data I want to generate. So in case of QTL-seq, it's nothing but it's a WGRS for 3 sample. Okay. So we'll be at least generating X data, at least 10x coverage. So suppose my genome size is 4.2 MB, okay, 4.2 GB. Lentil is there, so I'll be generating at least 42 GB data. Okay, so if I'm doing that, so this is called as like WGRS kind of a strategy, which is considered as whole genome resequencing. If I'm going with only 1x data, so that is considered as your GBS, genotyping by sequencing. Both the strategies are better than that of your SSR marker or any other PCR based marker. Okay, so for that we'll be using this QTL-seq or bulks. This is also called as BSA for QTL mapping. Then another term is your QTL confidence interval or support interval. Suppose I am mapping any QTL. So what we are looking at, we are looking like this. Always there comes a peak. It could be like smaller peak, a bigger peak, some kind of peaks are there. Okay, so this is what it is. Like different peaks are there, and then we'll be going with the flanking marker. So these are the flanking marker, and then we are saying that my QTL is sitting there. So this is being identified based on that. So suppose if there is a peak like this, so this is one QTL interval. This is my QTL position for green. This is my QTL position for red. So this is how we identify. So rather than going with this bar again and again, we present, like this peak again and again, we present this as a bar. So this is a pink bar, like green bar, and this bar. You'll be seeing a lot of papers like showing this kind of bar. Okay, so bar is nothing but it's your QTL position is represented as a bar, and then if same region will be having another QTL, so it can be presented as additional bar. Sometimes they overlap, sometimes they don't overlap, sometimes these bar are present altogether different locus. So length of bar is nothing but it is confidence or support interval. So what is your understanding? Length should be more or length should be less? I'm mapping a QTL, and one case I'll be having a bigger bar, another case I'm having a smaller bar. Which one is better?
Smaller one.
Smaller one, why?
Size will be lesser between the two markers. Our genomic region will be less. That will be better.
Correct. So confidence interval should be less. And the added thing is that if my confidence interval is less and then simultaneously my PV is high, so we'll be going with that. Suppose if my interval is less, PV 10%, my interval is high, but PV is suppose 50%. In that case, I'll be going with bigger bar, isn't it? So it's a combination of both size of a bar and then PV. If PV is high, bar is less, best. If not, so we'll go with the PV. So this is how we understand this confidence or support interval, and it extends based on the LOD score. This is a log of odd score, which decides this. So it's a LOD kind of a thing which decide that how this peak is going to be there. Okay. And then last, I'm going to stop over here. Then another technique is called as advanced macros QTL analysis. Okay. So what we do, QTL mapping is going on. I'll be mapping a QTL and then we'll be using in due course of time, as we have seen in case of Dr. Rajiv Varshney's paper. Okay. So, but it was decided that it was thought of that while mapping, if I could identify some good line which is having my trait of interest, so why cannot do that? So this is this methodology where we'll be making backcross, which is in advanced generation, doing the QTL analysis, and it is being used in the breeding program. So this is called as AB QTL, in nutshell, very, very basic. So Tanksley and Nilsson, way back in 1996, they devised this kind of analysis, which is called as AB QTL analysis, advanced backcross QTL analysis. So here we combine QTL mapping with breeding. Okay, so these two are combined. It's not like that we'll be first identifying QTL and then using them. So using, in this case, we are mapping population, backcross mapping population is being used, and then we'll be doing this analysis in BC2 or BC3 stage. And then simultaneously we'll be mapping also, and then whatever lines that where we found that, okay, this is having very good, means all my QTLs are there. So we'll be identifying those lines in case of my QTL analysis, and it will be used for breeding as a breeding line, or also it can be used as line for straight away release of a variety. I do have some more slides, but I feel this much is more than enough for today's part. Anybody who is interested, like to know anything more, that can always be dealt personal level. Okay. So with this, I would like to thank you all, and whatever questions you are free to ask. Thank you. So I'll be stop sharing. Okay, Dr. Jyoti.
Oh, okay, sir. Okay. So participant may ask few questions. They can raise their hand and ask, or they can have question in chat box. I'm seeing one question in chat box.
Hello. Hello, sir.
Yeah, tell me.
Myself, Dr. Ken Gupta from Jabalpur, sir.
Yeah.
Sir, any difference between mung bean yellow mojic virus and soybean yellow mojic virus and urdu bean yellow mojic virus?
See, if you see the reports, there are reports where MIMIV is identified and the same virus is affecting all these three. Okay. So we have one like construct is available there with me. So that we have tried in other cases, other crops also, but it is working very well only with the mung bean. It is not working that good with the other one. So these viruses, if you see, just like in case of plant system, in case of other system also, these viruses are constantly changing. Okay, but there are infectious clones available, which is equally good for all three crops. Okay,
right.
So that's the only reason MIMIV is called for all these three. They are only saying because it was first identified in this class.
Yeah. Thank you very much, sir.
Thank
you
you
very much, sir.
Thank you.
Sir.
Yeah.
So till date, there is no any report of release of variety in wheat
Mhm.
transferring the QTLs, and what will be the way out for this?
No, it will come in due course of time. See, it started with the smaller genome size, and then as I told now, identification of QTL is very easy. But then unless until you have a major QTL with you, and then genome, you see A, B, D, there are 3 genomes in case of wheat. So, and the genome size is a bigger constraint, but still now I feel the genomic is so good in case of wheat, in due course of time should be coming. Okay, in case of chickpea, again, they are very lucky that multiple QTLs are sitting together. Same happened in case of your groundnut, where like your trait of interest is being governed by just one major QTL. So it becomes easy to transfer. If suppose trait is being governed by 10%, then how many you are going to stack in one line? So it's again like trait of interest and then transferring, and then it will come. Like 5 years before, we are not thinking of getting any variety which is biofortified, but nowadays we are having a lot of varieties which is biofortified and based on even markers. So it's only like time and then complexity of a genome which is more important.
Okay, thank you, sir.
Yeah.
So I think you can ask sir later on also. We can share his email ID, or it is available on ICR website also. And you can ask, or you can even talk him for any queries, any clarification of dot. I would like to thank Dr. Gyan Prakash Misra, so who has vast experience of serving in different districts at ICR, DRDO, and at that UC Purdue lab, and he has worked on several other various crops. So certainly our participant must have enjoyed today's lecture and must have gained enlightened by the topics. So all the dots might have become clear. I have seen several messages in the chat for him. Very good message that all the things are clear. There is no confusion about genes, QTLs, mapping population, QTL spots. So thank you, sir, for your wonderful lecture and your time to give this wonderful lecture to the participants.
Thanks a lot. Actually, I always enjoy interacting in front of you, but there is no way out. It is online, so this is the only limitation for this lecture. Otherwise, really enjoy. Thanks a lot.
So certainly, so sir is we can say that he is the Mendel of modern India, great India. So he's a great geneticist. He was my senior at IRA, so I have learned a lot from him, and now also I am learning. So I have also enjoyed his lecture today. Thank you, sir.
Hello, ma'am.
Yeah.
Yeah, tell, please.
Please arrange one more lecture of the Gyan sir.
Okay, sir. Tell me
the topics.
Okay, if you will spare time, I will request him, because he agreed for Saturday being the holiday. Otherwise, he is very quite busy man. So I will request him if he can spare time. So
this is also a request from my side that association mapping is left. So it will be better if sir can spare some more time.
Okay.
We have another lecture on association mapping. But if I will request sir if he can spare on some holiday or some extra hours.
Yeah, it will be pleasure. Okay, so it will be like, what is the last date for you for this training?
It is up to 21st, sir. 21st.
Then we have a lot of time, huh? So
Okay,
some
sir.
odd hours, huh? Because this is constantly I'm teaching this. So for me, there's nothing like to prepare anything. Okay, if you give me like odd hours, it becomes good for me. Because I don't stop in 1 hour. So if you give me 2 hours, no, then this QTL I would have finished today itself.
We are ready to spend 2 hours for listening your lecture.
So association mapping, some odd timing, not on this regular part, I'll be taking surely. Okay.
Okay, then.
And
okay, we'll try to arrange. Yeah.
Then you can have better connectivity between these two. Fine. It will be more than it's pleasure for me to do this.
Okay, sir. Okay, so thank you, sir.
Thank you. It was nice interacting with you all. So I'm leaving now. It's fine.
Thank you, sir.
Fine.
Thank you very much.
Thank you. Thank you.
Thank you very much.
Thank you, sir. Thank you very much.
Okay, so thank you one and all. So everyone might have enjoyed the today's lecture. So now we have another lecture. So that is also very interesting. So today we have one very eminent speaker from our institute. The topic is very different. So, ma'am, I welcome you. Dr. Archana P. Raina is with us. She is a principal scientist in Division of Germplasm Evaluation Division, and she has also pursued his career from the IARA. She did PhD from IARA in the Division of Biochemistry long back. So she is principal scientist in the Division of Germplasm Evaluation, and her major area of research is phytochemical evaluation of medicinal and aromatic plants for quality traits. So as many of you are working on the medicinal plants, so you will gain several knowledge from her lecture, and you will have to satisfy your queries with her presentation. So Madam has more than 26 years of experience at NBPJ, as well as prior to here, she was at Simla, that Potato Institute. She has published more than 121 research papers during her career, and book chapters, and also technical articles. She has registered 9 genetic stocks in medicinal and aromatic plants for quality traits. Then she has developed various analytical methods for quality evaluation of active compound in germplasm of several important medicinal and aromatic plants. Based on, she's working on advanced techniques of that phytochemical analysis like HPTLC and aromatic profiling of crops by GCMS techniques. So you can visit to NBPGR also, you can visit her lab. Her lab is very much equipped with the advanced infrastructure facility. So ma'am, I welcome you for the presentation. So Rabi.
Thank you, Dr. Jyoti.
Yeah, so please share your slide, ma'am.
Okay. Am I audible?
Yes, ma'am, very much audible.
Okay. Good afternoon, everybody. And just, I'm sharing my slides.
Yes, ma'am, it's visible.
Is it visible?
Yes, ma'am, slide. So
Presentation mode.
Ma'am, presentation mode, you make double click.
It is not in presentation.
no, ma'am, presentation mode mein it is not there. Double click.
Okay, I will do it. It is presentation mode now?
No, ma'am.
मैम पहले प्रेजेंटेशन ओपन करके फुल स्क्रीन रखिए, फिर इसको आप शेयर स्क्रीन करेंगे मैम.
अब आ रहा है प्रेजेंटेशन मोड में?
मैम प्रेजेंटेशन मोड में नहीं आ रहे मैम.
But I put it on the slide.
मैम पहले प्रेजेंटेशन सारी ओपन करके रखिए मैम, फिर शेयर स्क्रीन
ओपन करके ही है.
यस मैम,
ओपन करके ही है प्रेजेंटेशन तो.
We can continue in this mode also.
If it is visible, I can.
Yes, visible. You can continue, ma'am.
You can continue, ma'am. It's visible. Otherwise,
it is visible to participants.
Yes.
Okay.
Yes.
Okay. Good afternoon, everybody. I shall be talking about phytochemical evaluation of medicinal plants. Today's talk I have divided into 3 aspects. In brief introduction about the medicinal plants, their quality traits, and what are the techniques we are following, and our studies, results of our studies, I shall be talking. Now, as you know, India is rich in biodiversity, and it is one of the amongst the 12 mega biodiversity countries. Around 80 to 90% of 50,000 medicinal plants are used, collected from the wild. Hello?
Slides are not moving, man.
Slides move.
Voice nahi aa raha hai?
नहीं, slides are not moving, ma'am. वो डबल क्लिक करके मैम स्लाइड पे डबल क्लिक करके फिर कर्सर डाउन
मैनुअली मूव करिए मैडम, क्लिक करके सेकंड स्लाइड पे।
Otherwise, you can send to me. I will update from here. अगर देख लेती हूँ।
नहीं तो फोन से शेयर कर सकती हूँ, ऐसा कुछ नहीं है। अभी आ रहा है?
हाँ, now moving.
Okay. Now it is moving?
Yes.
Okay. So okay, there is a gradual increase in medicinal and aromatic plant use in Ayurvedic treatment, in the Unani, as well as in earlier, there was in our Charka and Susutra, different plants were used in medicinal and Ayurvedic treatments. And coming to our phytochemical evaluation, what are phytochemicals? These are the plant compounds which are responsible for they are in addition to the essential nutrients. They are secondary metabolites. What are secondary metabolites? These are the compounds like flavonoids, essential oils, or anthocyanins, which are not required by the directly, but they are required by for other functions like defense against various predators, parasites. So there are two types of metabolites in plant metabolism: primary metabolites like amino acid, lipids, etc., which are required by the plant for normal growth and development, whereas the secondary metabolites, which are not directly involved in this process, but they are important for the ecological functions like defense, color, etc. So phytochemicals are just natural substances present in the plant, and they have activity like they are and having high antioxidant activities, which are gaining a lot of importance in today's time for prevention of disease and anti-aging. Now, these phytochemicals mainly fall in category of terpenes, alkaloids, glycosides, flavonoids, and saponins. This is the different classes of, my slides are visible to you?
Yes, ma'am.
Yes.
Yeah. There are mainly phenolic category, then terpenoid components, then nitrogen compounds like alkaloids. These are the nitrogen compounds which are present in the plant. Then there are carbohydrates and macromolecules like protein, sugar, nucleic acid. The phenolic compounds, they constitute flavonoid, anthocyanins, tannins. Similarly, in terpenoids, there are essential oils, then carotenoids present, like in even in the tomato, the lycopene is one of the carotenoids, which is having high anti-cancer activity. Similarly, anthocyanins, they are very important group of compounds. So this is just to show the type of chemicals present in plants. Now, what are the importance of these medicinal herbs? Why they are gaining a lot of importance these days? Because nowadays we are moving towards the herbal plants as they are not having any side effects, and they are used in the drugs, pharmaceutical industry, then as a nutraceuticals, like functional foods and designer foods, in health drinks, then in cosmetic industry, like you all know, aloe vera, which is having a lot of products for in the cosmetic. Similarly, biocontrol agents like plant extracts and essential oils, they are used for the disease prevention and other activities also. So therefore, these plants are gaining a lot of importance, but there are a lot of challenges in medicinal aromatic plants due to they are not being cultivated by the farmers, and they are being harvested from the natural populations, which is causing loss of species and habitat. And there is a technological gap because there are no agro techniques or standard techniques of operation for these crops till date standardized. And there are not high yielding varieties like you are having varieties in common crops like wheat and other important crops. And therefore, these techniques are required to be standardized. These are the limitation in these crops. Then there is limited information on genomics as well as the secondary metabolite, biosynthesis, then even the quantification. Now we don't have the, earlier we don't have the standard methods for quantification of these compounds for evaluation of medicinal plants quality. And due to which there is a quality standards are not established so that the industry can get a uniform material, quality material. So there is non-availability of standardized production as well as the processing technologies for these crops. The lack of post-harvest technologies and value addition for better shelf life, quality and target delivery, and then lack of economic and efficient technologies for extraction, characterization, and isolation of bioactive molecules for high value standard products. So these are the main issues which need to be addressed. Keeping in this in mind, there was a National Medicinal Plant Board was formed, and it is headquartered at Delhi. And initially, they identify 32 main medicinal aromatic crops, which can be, which were promoted for the cultivation, development of agro techniques. And some of the crops, these are very important, like aconitum. Then these crops with asterix, these are some of the crops are found in high altitude and they are difficult to collect. Like aconitum is a high altitude plant. Similarly, then there are Endographis paniculata, Kalmic, Bacopa monri, which is a nerve tonic, commonly used for memory enhancing purpose. And then Asparagus shatavari, which is a health promoter. Then Acacia senna, it is one of the very important crop, seeds of which are having senosides, having high purgative activity. This is the exported for the plant. Then Chlorophytum, which is you must have heard, Safed musli. It is was very, very expensive and it is health promoter. Then Coleus, then our Amla, that is also having high medicinal activity. And then Glorisia and our saffron, then Mulhatti, which is called Glycyrrhiza, and then Nadostachys jatamanasi. This is another important crop, which is having valioprates and good for the nerve tonic. And similarly, Picorrhiza kuri, Kutki, it is one of the endangered plant found in hills. And Plantago ovata, commonly taken as isabgol. And you must be familiar with this isabgol's importance. And then Rauwolfia serpentina, Rasarpi, is the main compound present in this plant. It is commonly called as Sarpagandha, and it is used for high BP. Similarly, the Solanum species, Antinospora cordifolia, during this COVID time, you must have heard people were taking giloy extract, the Tinospora stem extract for increasing immunity. Similarly, Withania somnifera, Ashwagandha. These were the 32 initially identified crop by Medicinal Plant Board. and they started promoting these crops. Later on, now they have increased it to 120. So different parts of the plant are used. Maybe it is bark, stem, seeds, depending on the active compound. So somewhere whole plants are also used in the industry. So these are the like Eagle Marflorus and then Plantago. They are used for diarrhea, different. Similarly, Bacopa. Aloe vera is used for the skin. And our haldi, which is known as Curcuma longa, it is having high anti-inflammatory and it has anti-cancer activity also reported. And this is chlorophytum. And Terminalia, this harrod and all, good for digestion. So some of the aromatic crops like eucalyptus oil, then mentha, similarly Cymbopogon species like lemongrass, Pomarosa, these are also essential oil are used in the medicinal purpose. In most of the active compounds, like in papavara, which is poppy, morphine is the active compound, codeine, which is used in the cough syrups. And it is, although it is a banned crop and it can be only grown by taking permission. Similarly, Cinchona species, they are having quinine, which is anti-malarial compound. Digitalis having digoxin, which are the cardiac glycosides. And then the Scaturantha species, you must have seen at your home, Sadabahar ka poda, having purple and white color flowers. Now the leaves as well as rhizomes, roots are important for the active medicinal compounds. Now the leaves are having vinblastine and vincristin compounds, which are anti-cancer. Now these are the medicinal plants at our Delhi center, maintained in the gene plants. Hope you are able to see the plant pictures. Vetiveria, you must have seen roadside pe bhi kabhi kabhi it is visible, having it not only having a property for this perfumery and all, but it is also used in our coolers for the cooling purpose, as well as it prevents the soil erosion. Similarly, Pomarosa, it is used in the soap and perfumery industry. Aloe vera. So all these plants are maintained at our farm. Whenever you get opportunity to visit our NBPGR, you can visit farm. There they are maintained in the field gene bank. In addition, some of the crops based on their requirement of climatic condition, they are maintained at our regional stations, like some of the aromatic crops like rosemary, oregano, they are maintained at Mediterranean crops. They are maintained our Bhowali center because they require the cold temperature. Similarly, our Trichur center has plants which are found in the Western Ghat region. They are having collections of Alpinia, Bacopa, Campharia, different medicinal plants are found there. And in our eastern areas like Cuttack, they are having Ocimum, Gingiber, and Hedychium species. And in northeast, we have Shillong station, where they are also having Alpinia, different Curcuma species, Perilla, they are found in the northeast Shillong station. So these plants are difficult to establish. So whenever our explorers are going for collection, and so they need to be established to the climatic condition where they are found, so that the quality traits don't change. So last lecture was on your mapping of genes and QTLs. So this topic is totally different here because we shall be mainly concentrating on the quality traits, which are very important in these crops, and economic importance of these crops are from the quality traits. So what are the parameters required for the standardization of these drugs? It is very important since these drugs are not cultivated. So one has to see the correct taxonomic identification, authentication. Most of the people are getting from the forest or any, and since the quantity of active compound is less and the material is also less, so people are adulterating also. So there is requirement for taxonomic identification, then study on different parts like root, stem, bark. So that is also very important. Then harvest stage, and then organoleptic evaluation of the crops by touch and odor. And in the chemical studies, we are doing TLC, GLC, HPLC, as well as the DNA molecular studies by fingerprinting. And evaluating biological activity of the plant is one of the very important parameter, and shelf life of the drug. So why we need to study the quality parameters? Because they are affected by the climatic condition, genetic factors, then different part of the plants have different compound, and then conditions, culture condition of the plant, where the environmental condition, then development of the plant and plant stay parts, as well as the post-harvest management of herbs, and then method of extraction. I don't know if anybody is working in this group on the medicinal plants, so they will better be able to understand these, why these parameters are important whenever we are studying. So this is just to show the what are the main things, microscopic analysis by scanning microscope and all, before going for the drugs for the industrial utilization. Then HPTLC and GLC and HPLC, that is the final stage where one goes for the final chemical evaluation of the drug. These are required by the industry while giving out the drugs. So medicinal plant, they are also very important. They should activity evaluation, like medicinal extract. They should, they can be used for bioassays. Then one can go for the purification of the compound from the extract, and then identification, structure elucidation, and then toxicological studies. Coming to the different extraction, here we have ultrasonic extraction. So extraction procedure is required to be standardized so that active compound can be isolated. Then oxalate extraction is another method. In case of aromatic essential oil extraction, can be done by hydrodistillation or steam distillation. Then microwave assisted extraction is the nowadays recent technology, and supercritical fluid extraction is an accelerated solvent extraction, but they are very costly technique. So they cannot be followed at the field level. Only on the lab level it can be done. So this is the most common extraction technique used for essential oil extraction, that is hydrodistillation. When plant material is put in directly in contact with the water and it is heated in the lab, we have just shown that small heating mantle can be used. When you boil the sample, the oil comes from the sample, comes along with the steam or vapors. It is then condensed by cooling, and then that oil can be collected. Now one has to standardize the technique for different plant material, the essential oil, how much time is to be given for hydrodistillation. Then this oil is collected, and this oil can be further analyzed by gas chromatography, where one can use the particular column. These columns are specific for the compound to be separated, and one can get the chromatogram, where you can get the list of compounds which are present in the oil. And it can be used detector. Detector will give you the what are the chromatogram. And generally FID and MS, these are the most common detector used. MS is to identify the molecule based on mass to charge ratio, where the sample is iron, you will pass through the ion source, and mass analyzer will give you mass to charge ratio. And here you can identify the unknown compound also. In these plants, there are, since the number of compounds are more, so MS is very useful to identify the unknown compounds. Now, this is our lab view. Whenever you get opportunity to visit NBPGR, you can see this is the GCMS facility. You have the auto sampler, and in these vials, we analyze the by the sample. And then another important technique is for this GC is used for the volatile components like essential oil. And if the compound is not volatile, then it has to be derivatized. That means it has to be made volatile. Another technique is HPLC, where it is used for the non-volatile component using HPLC's high pressure liquid chromatography. Maybe many of you must have used during your research. And there you can separate the compound of your interest based passing through the column, HPLC column, and detectors like PDA and RI detectors are commonly used to separate the compounds. Now, this is another important technique, that is HPTLC, which is high performance thin layer chromatography. Here the compound is, it's like simply like a TLC, but here things are in automatic mode. The auto sampler will load the sample. You have to make a program and then it is scanned through the scanner. And through the scanner, you see this is the spectra of the compound, and where you can find out that this is the maximum absorption of the compound. You can identify whether this is the compound of your interest. Or when you have the standard in herbals, there are difficulty of getting even the standard. So by this technique, even we can separate the compound and then get it identify using NMR and these structure identification. That is the thing which we can isolate the compound, because reference compounds are not available for all the medicinal plants. So we need to isolate also sometimes. And then isolation, one has to check the purity of the compound. So that is done by the characterization using NMR, ioscopy, advanced techniques. So this is HPTLC. Coming to our studies, so we have Endographis paniculata germplasm at our Isa fruit plant. So this herb is commonly called as kalmeg or green charaita. And this is found in throughout India, and whole plant is used, but different parts contain different composition that we studied. What is the variation in the endographolite content? This is used in many of the drugs used for the liver disorder, like LIV52, it is one of the component. And it is also used for the fever. And nowadays, its activity for HIV also reported. So this is very important plant. And this is the, it increases the number of usage, like it promotes digestion, then liver and hepatoprotective effect, and many uses are there reported for this plant. So here you can, I hope you can see that different collections from different parts was collected from 14 states of India. And the plant, then they evaluated the endographolite content, different parts of the plant, like leaf, stem, and capsule. Here you can see the TLC picture on one side in green color, where you can see that in the last there are the standards loaded, and in the first sample loaded. So corresponding to standard, we identify the endographolite in our plant sample. Then in the different parts of the plant, we could see that leaf color in blue. You can see the bars color in blue. They are showing the maximum endographolite, that is from 1.32 to 2.60% for the in the leaf, followed by the stem, where the capsule was having very less content of endographolite. So that means it is mainly the leaf, but in the industry or the people are using whole herb for the cure, but it is the leaf which is having main component. And then we could also identify the areas like Madhya Pradesh region, they were having, and plants from collected from that area were having high endographolite. So another important plant was the chiraita or the savartia, which is commonly called. It is found in hilly regions like Himachal Pradesh, Kashmir, Meghalaya. It contains swarshins like alkaloid. So this is also good for stomach and anti-inflammatory and skin diseases. And generally it is mixed in place of kalmeg. Adulterants, I was telling you, that sometimes the material is the people are substituting also. Then another crop which we studied was, this is the diversity you can see in Mucuna pruriens, which is also a leguminous crops. But tribals are also eating after removing the water. It contains L-dopa, which is a derivatized amino acid, phenylalanine amino acid. So this is the variation you can see in different species of mucuna, that is Gigantia, Mucuna pruriens, and Monosperma. So here you can see the white seeded mucuna was having less L-DOPA, whereas the Mucuna pruriens, which is very commonly found, was having up to 7.60. One of the accession was very good, so that we have also registered for the genetic stock. And then this is our Ossimum holy basil. This is almost in all the houses in India is worshipped for the also for, and the leaves, it is having two morphotypes, that is Shama Tulsi and the green Tulsi. It is used for common colds, headaches, stomachs, then heart diseases. Essential oils are usually extracted from these plants, and we could get two chemotypes in this species. Chemotypes are basically typing of the germplasm based on the chemical variation in the chemical composition. So eugenol, one was eugenol rich, other was methyl eugenol rich. Recently, we have also found other chemotypes available in our germplasm, which were rich in beta caryophyllene, high in beta caryophyllene, and one was beta caryophyllene aluminine rich also. So we have large diversity as well as germplasm also. We have around 150 accessions of Ossimum sanctum. Then we have other species of Ossimum also in our gene bank. This is Ossimum basilicum. This plant is used in not all in other countries like US and all as a flavoring and as a culinary herb. Now this basilicum is having different chemical composition. So this helps us that we can have the chemical based on the species. So chemotaxonomic markers also. So different species can be identified based on the chemical markers. So here we don't get eugenol, methyl eugenol as was in Ossimum sanctum, but in Ossimum basilicum we get different compounds like linalool, methyl cinnamate, methyl chavicol. So these were the three main chemotypes which we found in basilicum species. Now, this basilicum from the eastern region was found having methyl cinnamate rich chemotype, whereas the chemotype like methyl chemicol or linalool was available in this Delhi region, Ossimum germplasm. So here this graphs, I hope if you can see the two in the first graph, there are linalool and methyl chavicol. They are the two major compounds present in high content. In the second graph, linalool is the main compound. In last graph, the methyl chavicol. So these are the three chemotypes which are identified in the germplasm. And the first one, that is linalool and methyl chavicol, is the most preferred chemotype. It resembles to the European type of basil, and it is highly in demand. Now, another important plant which we studied, that is Kaempferia. It is found in the southern parts. We got this collections from Western Ghat. So this plant is good for the expectorant, diuretic, as well as the extract. Rhizome extract is used for the joint pain, and essential oils are present in the rhizome, which is rich in ethyl cinnamate, camphor, borneol. Here you can see this is the graph which shows that ethyl methoxy paramethoxycinnamate was the major compound in the germplasm, which we tested, and then ethyl cinnamate and pentadaconium. Now this ethyl trans-paramethoxycinnamate is very important compound for anti-tuberculosis, and it is being used for the studies for developing the drugs. Now this aloe vera, this plant, I think most of you must be very much aware of this plant using. Now the plant leaves are very succulent, having gel, which is used in the cosmetic industry. Not only that, the plant contains a compound like aloin, and which is a yellow type of solution coming out of oozing out from the leaves. This is having good activity for the digestion, and there is a company Forever named Forever, US company. It is developing products more than 900 from single plant of aloe vera to dandruff shampoo, then face creams, moisturizer, and single companies having more than 900 products from aloe vera. So this is a very important crop. Aloe vera juice is nowadays very commonly sold in the market, and this aloe juice is having aloin. Then the sports persons, they are having drinks from the aloe vera because the juice, aloe gel is rich in the carbohydrates and amino acid. So that is why the health drinks are promoted from this plant. So this is commonly called as Grit Kumari or aloe. It is found in barbolene or aloin. These are the two main compounds and good for the stomach ache. So this is another plant, that is Bacopa mondri. This is Brahmi, and this plant contains bacoside A, B in the leaves. So this plant is used for improving nerve, this memory problem and nerve tonic. And recently we evaluated the Bacopa germplasm available at our station. So up to 3% we can get the bacoside. That is quite a good amount of bacoside present in the leaf. This is the Tinospora cordifera plant. It is a basically climber and it is growing on a tree. You can see the stem of this plant are used for the drugs. Stem contains tinosporoside, cordiofolioside, and this plant is gaining a lot of importance. And you must have seen in the Yoga Guru Ramdev is a lot of promoting this plant, and now this giloy stems are also sold in the market. Stem is the powder is used for making the extract or decoction, and it is known to have effective for the fever and immunity enhancer. Now this is the senna plant I was telling you. The seeds are very important. It is having pods, and then seeds are having senoside A and B, which is used for purgative, and these are exported, and India is one of the main suppliers of this senna as well as the plantango. This is Withania somnifera. Now this has rhizomes are having alkaloids, withanaferin or withanaferin A and B. This is the ashwagandha commonly called, and it is a tonic. Now this is sarpagandha plant, and the rhizomes and roots are having reserpine alkaloid, which is good for high BP. This was the first plant which was identified for this problem, medicinal plant. Now this is there are different species of sarpagandha, but this tetraphylla, these are the main species, and roots are used for hypertension. This is Sadabahar I was talking to you. Now you must have now recognized this plant, the Catharanthus roseus, white and purple flower. Now leaves are very important for vinblastine and vincristine alkaloid used for the anti-cancer drug. Similarly, roots contain agmelicine and serpentine, which are all for good for the high BP. Now this is Centella asiatica, another important plant for the memory enhancer. Now this is Piper longum, good for use for the coughing and bronchial problems. This is plantago and plumbago. Now this is faith musli plant, Chlorophytum borivilia. So this plant is roots are having saponins, and it is used for health promotion. Now, similarly, sattavar roots are also very important for health promoting asparagus. Now, this is mentha. Mentha, as I told you, this is oil is used for the medicinal purpose. Even the oils are used for in our toothpaste and also. Then another important crop is lemongrass, Symbopogon fluxus. Now you can see that this plant contains citral A and B, which is a precursor for the vitamin A synthesis. In addition, you must have heard lemon tea or this plant leaves of the plants are having smell like lemon. So they are used for preparation of herbal tea, and this is a very important plant. And then vetiveria, this is Pamarosa. Pamarosa, Rosha grass commonly called as. Now the Pamarosa oil is very good flavorant and it is used in the soap and shampoo and all other industries, toiletries. Then this is the vetiver plant. Now you see the roots. These roots are used in the cooler as the gas, as well as the roots are rich in essential oil. So essential oil is having compounds like cucinol and all. This is a very important oil for perfumery industry. Now this commonly drumstick or Moringa oleifera. Now the leaves are rich in the flavonoids, and whereas the seeds are rich in the oil. This is the high oleic acid rich oil isolated from the seeds of these plants. Moringa is having number of medicinal properties are reported from the leaves. So people are even cooking these leaves for the vegetable purpose. Now this is the our spices are also, that we are having India's people are taking lot of spices. They are rich in active compound, and the compounds are known to give the medicinal properties like our curcuma, haldi. This is having curcumin. So these are all giving the medicinal properties. Similarly, coriander seeds, they are rich in essential oil, having which are linalool rich oil. This was the germplasm evaluated, and we could identify high linalool content in the seeds. Now there are some of the accessions which we identified for the high sorrelline content, like in Sorelia coralifolia. So the seeds were having high sorrelline were registered. Similarly, Alpinia species having high cineol content. Now, this Alpinia species is used for the bronchial problem, and so they were found to have up to 72%. Similarly, Calcareta, another species of Alpinia, it was having, in addition to cineol, the fenchyle acid. So that germplasm evaluated, and we could identify the lavender stoicheus. Now, there is two species of lavender. One is lavender used for the perfumery, rich in linalool and vinyl acid. This was the pharmaceutically important species having high camphor content. So this high camphor content was very important for the pharmaceutical industry. So this species were identified. Then origanum, those works I have actually skipped some of the slide. That we also evaluated origanum germplasm from our Bhawali station. So we could identify that Origanum vulgare, which is this seeds are used also in the pizza spice basically. So they are rich in the phenolic compounds like thymol and carbachol. So we identify two collections. One was high in thymol and other was high in carbachol. So these two compounds are very important as they increases the shelf life of the product, as well as good for other medicinal purposes, antioxidant activities. And Osmum basilicum, we have identified high methyl chavicol rich germplasm. And in Mucuna pruriens, we have identified high L-DOPA content. And in Osmum basilicum, we have also identified high linalool rich germplasm. Now, there are a lot of benefits of medicinal plants. The time has changed. Many people have realized also. I've seen many people coming in, have they have started cultivation of these plants on the commercial scale also, because they are required to be cultivated for supporting the modern medicine industry and getting authentic raw drug material from the cultivator. Then also export of raw drug material and extracts. Now there is another important plant, that is Garcinia. Now this Garcinia is found in those who are from the South India. They must have seen many species of Garcinia. They are Garcinia indica, Garcinia cambrogia. This plant, that fruit rind is rich in HCA, that is hydroxycitric acid. This is an anti-obesity plant. So this this fruit extract, they are being exported to the outside India. So this is how the people are moving. And similarly, some of the essential oil industries have been set up in this Chamoli and Bhopali region. So there also people are sending the oils from here to different countries. Now, not only this, these plants are very important from the point of view of utilization of fallow and marginal land. Some of the plants are very hardy, like Bithania, vetiveria. These you must have seen. They are found in the growing in just adjacent to roadside, like Adothoda, another important crop for the bronchial problem. So they can be grown in any type of climatic conditions, and they don't require any much input, and they are because of these compounds, that is additional advantage imported to these crops. They don't have any disease problem, so they don't require many type of efforts by the farmers, so they can be grown. Similarly, some of the crops they can be used to prevent the soil erosion, like you have seen vetiver growing all around the areas. So and another is employment generation. So these, since these crops can be small area, they can be, people are growing like chlorophyta. Many people were getting lot of money by just growing a small area, because they are, earlier it was sold at the cost of rupees 1,000 per kg it was being sold. So the employment generation as well as the money generation people are doing. Then also by growing these plants, we can conserve the biodiversity, because by cultivation, people will start, our natural resources will be not depleted, and people will start going them. And then these can be grown in agroforestry, like intercropping. Some of the forests here, some crops can have been, people have started growing those, so that whenever the farmer can utilize that loan. So therefore, and now the funding has also started coming from different institutions like medicinal plants, many other organizations. They are promoting cultivation of these plants for the area. So that's all I wanted to tell. And if you have any queries, you can ask me. I'm sorry, I think the slides were not properly shared, I guess.
No, that's fine. That was very much visible. The slide was very much visible now. Okay. So now the participants can ask a few questions. So Madam can take up a few questions. Or they can put the question in the chat box.
Anybody who is working on the medicinal plants or aromatic in this group, because this is an online, we don't know that what is your area.
Yeah, some are working. Yesterday we took the introduction of the candidates, trainees, and some of them are working on the medicinal aromatic.
Okay. Because it's a totally different aspect. If somebody is working, then they will.
Hello.
Yes,
Hello?
please.
Yes, ma'am. I am working with medicinal and aromatic plants, especially to standardize the production technology of the medicinal and aromatic plants.
I know your center having lot of work on that area. Earlier, Dr. Mishra was there, na?
Yeah, ma'am. Yeah. Dr. S.N. Misra at Mansoor. Yes, ma'am. Yes.
So what?
Nowadays, ma'am, I have also started crop improvement program of Satawar, Gudmar, Sugandha and Sarpagandha. So I am collecting germplasm and hello. Now I have started to collect some useful germplasm.
Which germplasm?
Germplasm of Satauri, Gudmar, Sarpagandha, Sugandha and beetle white.
Yes. You have tissue culture lab also, no? There?
No, ma'am. No, ma'am. Here at Jabalpur, I have a tissue culture lab, but not a well furnished.
Okay. No,
And
Jabalpur is having good.
sometimes it is very difficult to analyze the bioactive compound of that medicinal plants. Until
Yes.
and unless we analyze the bioactive compound, we cannot say this is a very useful plant or not. जब
That
तक
is true.
बायो कंपाउंड पता नहीं चलता तब तक हम किसी जल प्लाज्म को
हाँ.
अच्छा या बुरा कैसे कह सकते हैं?
Yes, that is true. And that is very important for the industry point of view, bioactive compound.
Yes, ma'am. Yes.
And then they are present in very small quantity, so it is very difficult also to quantify till you have this good method, standardized method.
Right, ma'am. Right. And standardize, some chemicals are also very costly for the standardization.
Yes, yes. You have facilities for quality evaluation?
Yes, ma'am. At our university, a quality control lab is a quality analysis lab is there, but they want a standard from my side, and I have not money to provide him that standard. They are ready to analyze, but they ask for the standard.
Okay. No, you can
ma'am,
send me email.
If I send you some material to you, you help me or not?
Yeah, sure. You can send me mail depending on the crop. I can let you know, because we are also not working many of the crops which you mentioned. But I can, if I have, I can help you definitely.
Okay, ma'am. Okay. Thank
You can take my email address and you can mail me. No issue.
Okay, ma'am. Okay.
Hello, ma'am.
Hello.
Hello, myself Dr. D.K. Payashi, Devendra Kumar Payashi. I am scientist in charge AICRP on linseed. Linseed, flaxseed.
Yes, yes.
Ma'am, I have the fatty acid profiling of those varieties which we have released. We have released more than 10 varieties from our station, and now we need to analyze the micronutrient analysis, especially calcium, magnesium, phosphorus, zinc, iron, sulfur, which are very important in human body, because flaxseed is nowadays used as a food material. Then carcinogenic substances, whatever carcinogenic, cyanogenic is one of the carcinogenic substances having minimum quantity. Then fatty acid profiling. So these aspect, these quality aspect, I am interested to collaborate with you. We have already DBT network project with us. Under the coordinating unit of NBPGR, Dr. Vikender Kaur and Dr. Ashok Kumar sir is leading the coordinating this. So we have funds, and we are looking for coordination to analyze all those samples, released varieties, and their quality aspects, ma'am, which are essential for edible food.
Okay. You can do mineral analysis by outsourcing. ICP-MS is available many places.
Okay.
Because we don't have ICP-MS, and for the complete mineral profiling, you will be requiring ICP-MS.
Okay.
And there is, I think, a collaboration with the Jamia Hamdard also, no, for linseed.
Jamia?
Yeah.
No, ma'am.
In that project?
No, not at all.
Okay, they are actually working on some medicinal aspect.
MIT University is working with us. Dr. Nutan
MIT
is working for quality aspect.
Okay.
Or they are working on especially fatty acid profiling and oil.
Okay. Yeah, that is mainly unsaturated fatty acid, linoleic and
Yes,
linolenic
yes,
acid.
yes.
So For mineral, you can collaborate or outsource also, because in IRA also ICP-MS is there. If you want the complete profile of a mineral, we don't have ICP-MS.
Can you suggest me the name of the person whom I can collaborate in IARI?
Okay, I think Dr. Rakesh Bhardwaj's presentation is also there, no?
Yeah.
So for mineral,
his presentation is also there,
can,
detailed presentation. So he will be talking in more detail
yeah.
those aspects.
Right, ma'am. Right, ma'am. We will discuss.
So for mineral, you can talk to him. Otherwise, you can in IRA there is ICP-MS. I know.
In soil science
so you
division
can,
it is there.
Yeah. So you can collaborate, or it is in the market also. If you have funds in that project, there are outsource that industry companies are there who are doing it. I can suggest
Okay.
you. You can mail me. I can suggest you.
We have funds, but we cannot purchase and install at our station. We can pay for the analysis.
Okay. Then it should
Right,
not be a
ma'am.
problem. If you have funds, nowadays people are going for outsourcing because ICP-MS is 1 crore equipment. So it is very expensive.
Right, ma'am. Right, ma'am. Okay.
Okay.
Thank you very much, ma'am.
Okay. Okay. Thank you.
Okay, so any more queries? Any more question by the participant? One person, okay. Vinod Kumar, he has already talked, no? He has written in chat box, ma'am, how can get planting material of medicinal plant? So you can make the indent
Yes.
and
You
and
can write to
get the material.
NBPGR. And
Madam has shown several genetic stock also. So the person
Yes.
working on those crops can get indent of those medicinal genetic stock, and they can utilize in their breeding program.
Yes, you can write to NBPGR, whatever material, if they have at headquarter, or you can get it. That is not an issue. Then there is an we have another ICR Institute, Directorate of Medicinal Aromatic Plants at Anand also. It depends which crops you are interested. You can see their website, our website. You can write to us. You will get it.
Okay. Yeah. Further, if you have any queries, so means you can mail to ma'am, and we will be circulating the detailed contacts of all the resource persons to you. So you can have on phone or through mail. You can have the further discussion. So now I would like to thank ma'am, Dr. Archana Rana, for delivering such a wonderful talk, because now the time is very much health crisis is present everywhere. It is prevailing. So this medicinal and aromatic plant, this play very major role for this health crisis, as this is having several bioactive compounds which are useful for alleviating several diseases and health disorders. So thank you, ma'am. Thank you, ma'am,
Thank
for
you,
enlightening the
Jyoti,
participants
and
with your this.
thank you, participants also. They must be waiting for the lunch, and for thank you all. You can anytime email any if you have any doubts or any queries. Thank you all.
Yeah, thank you, everyone. So now your next class will be at 2:30 p.m. after the lunch, post-lunch session. So you can join sharply. That's a very good lecture by Dr. Sunil Archak. It is related to PGR informatics, PGR portal. So how will we go through the PGR portal? How will search the portal to get the information about the various important plant genetic stocks? Those things will be dealt, and he has developed several apps, PGR apps. So those things you'll come to know. So please join at 2:30 sharp. Okay, thank you.
The training program in this training program on acquisition, that is either exploration and collection from within the country. How do we import from other countries? How do we characterize large scale germplasm accessions? How do we evaluate in terms of traits, both field and molecular level evaluation? and then quarantine activities, conservation activities in different modes. So these are all very specialized activities. So you know that we carry out huge number of different activities, all part of the same management procedure. Now that we have so many activities, how do we manage the information that is required to efficiently take care of such activities? And how do we manage the data that are generated out of as a result of these activities? So this is very important for all of us, just in the morning, there was a lecture in one of the G20 series, and it has been declared now that it is the era of, it has been stressed over and over again, but in the G20, they have just mentioned in the morning, forenoon, that now is the era of big data. So what is big data in our area, in our field? Big data is anything related to the germplasm, the genotypes, the accessions that you're working upon. Okay, so I'm going to talk a little bit on that, and then I'll be very happy to okay. So the slogan or the purpose of PGR informatics is enhanced access to information for enhanced utilization. So this is no rocket science. If we do not have any information about a particular accession, a particular genotype, or your own breeding lines, from where they have come, what are the traits, and then you will not be able to use them efficiently. So that's the purpose. Okay. Okay. I come from NBPGR, the same organization which is organizing this particular training program, and we have all these regional stations. And then very proud of our organization. It is the second largest gene bank, and many other positives, a number of things. And if any one of you has not visited our gene bank, our institute, I request you to please come whenever you come to Delhi, come and visit gene bank. So is it the only gene bank in the world? No, there are so many gene banks in the world. You look at this map. This is the map of all the worthwhile gene banks. There could be some small, very small gene banks. These are sufficiently large gene banks where thousands of accessions are conserved. This is particularly about the XA2 gene banks, not on-farm into kind of gene banks. XA2 gene banks, mainly seed gene banks. So you can see how many gene banks are there all over the world. So many gene banks are there. So in so many gene banks, okay, where do we stand? Our NVP, I just mentioned, we are second largest seed gene bank in the world. Second largest seed gene bank in the world. And why do we say that? Because you can see here, we conserve almost all the crop species, because those very well-known gene banks such as IRI, IRI, Rice Gene Bank, CIMIT, ICARDA, there are a number of other gene banks which are managed by CG centers. These focus on a few number of species, whereas we work on almost all crop species and their wild relatives. So we conserve them and then we manage them, all kinds of activities. So this is a very huge gene bank, a set of mammoth number of activities, few people working on a number of species. So it requires a very, very efficient management of resources and information, which is why we need, again and again, I'm stressing, we need a very efficient database management. Okay. So what are all the activities that we carry out here? I think some of these are already explained in detail. On the left side of the panel, you can see acquisition, that is exploration, collecting, import. And because when we bring that one to our gene bank, we also carry out quarantine. When we are sending also, we are carrying out quarantine. And conservation through different complementary conservation strategies, seed, in vitro, cryo, field, DNA bank, all these. And then we have to use them. So trait discovery, so characterization, field evaluation, biochemical, genomic, lots of kinds of evaluation work is going on. So basically 3 sets of activities: acquisition, conservation, and trait discovery. So once we carry out all these activities, when I say we, it is NBPGR or any other gene bank. Once we carry out acquisition, conservation, trait discovery, they are available with us. But are they accessible? Question is, are they accessible? They are available with us in our gene bank, they are there. But are they accessible to the users, to you, to any one of you, anybody else, student, researcher? Are they accessible? So accessibility depends upon information. you will be able to get our material that is conserved in our gene bank with some specific trait only when you know that they are there and they possess these traits, and these are the conditions, or these are the methodology or the procedure through which you can access this material. So which is why availability becomes accessibility only through an efficient information management. So for that purpose, on the left side, you can see here what kind of total records that we are managing. One second, I will move this back one. Okay. We have developed these databases, and we are maintaining these databases, updating, carrying out all sorts of computer related activities for databases, for indigenous collection, exotic collection, conservation, all activities you can see here. I have just listed only 12 here. There are many other activities. And these activities at what scale? See, activities are more, but scale is also very, very high. What is the scale? You can see on the right side. NBPGR is conserving so many accessions, which are very, very higher than the other gene banks, CG gene banks, very well known gene banks that are conserving. So we are conserving huge number of accessions. Only USDA gene bank is conserving more than our gene bank, but they're not carrying out all the activities that we are carrying out. Okay, so that's also very, very important. So whenever we try to talk about PGR information management, there are very simple, fundamental, basic questions are there. Okay, why should we manage? Why we should manage information related to plant genetic resources? And if we manage, and what kind of information are there in this management? And how do we manage this information? And for whom all these things are managed? So if I answer these quickly, then we will go further. Okay. Yeah. We manage this because we want to manage germplasm efficiently. Okay. Our management of germplasm has to be efficient, so we manage information. And for whom we manage for three kinds of people. One, for all of you researchers who want to use these. One, for ourselves, for gene bank managers, because we want to manage. And the third one is for policymakers and funding agencies who provide us with the funds, who take care of our policy issues, who regulate us. For example, for we receive, let's say, parliament questions. They know exactly what are all the issues. So we have to give them proper information, which is updated, accurate, okay, and very reliable. So all these things are possible only when we have information systems. And then how do we manage? We manage based on a number of specific databases that we have constructed. And how this should be, there should be, I just mentioned, they should be very, very accurate. Okay, if I tell you that this particular accession is susceptible or resistant, then it should be, that should be true. If I give the information to the parliament saying that 136,000 land races of rice are there, so it should be true and correct. So they should be always updated, accurate, and reliable. Apart from all these things that are important for our own purpose, there are something else which are also very relevant. So I have to mention here. Perhaps Dr. Pratibha Brahmi has brought out this topic when she was presenting. India is a signatory of some of these very, very important international treaties or fora, such as the Convention on Biological Diversity, and then Global Plan of Action for Plant Genetic Resources, then International Treaty for Plant Genetic Resources of Food and Agriculture. In all these, it is mandatory for the signatories to ensure that you have an efficient plant genetic resources information management system for various purposes. I will not discuss them here. For different articles, they have ensured that these are minimum things that one should do. So India is one of the forerunners in this. We have excellent management systems. And what kind of information? As I just mentioned, it's a common sense to start with, but little more technical to end up. That we need information on passport, import, seed health, characterization, conservation, supply, regeneration, evaluation, utilization. So for each accession, these information bits will start building up. Once you bring it from, let's say, from wild, it has got passport information, from where it has come from, what kind of population it has been representing here. So all these things. The moment then you add acquisition information, you add trait level information, you keep on adding more and more information to ensure that its utilization is enhanced and let's say expedited. So that instead of using it, let's say 10 years later, you start using it right now in case of crop oil relatives, let's say. Okay, I'll skip it here. Yeah, this also I've already explained. Okay, now quickly, what is plant genetic resources informatics, PGR informatics? Why do I use this word? This is not a common word. After I have got ICR National Fellowship on PGR informatics, so that is where I have proposed this word, and I have been using this word everywhere, PGR informatics. This is the only center in the world where we are working on PGR informatics, and you should be happy that ICAR, Government of India through ICR, is supporting this kind of activity, specialized activity. So what is PGR informatics? PGR informatics is very simply management and analysis of diverse information that is related to plant genetic resources. So when I say management, there are again few different specific activities. That is creation, storage, retrieval, and presentation. That means something that I start with the people who are actually primary data generators, those who are explorers, those who are characterizing on the field, and from there I start creating, then storing through all standard globally accepted methodologies, infrastructure, okay, then pipeline. So these are all used for creation, storage. Once I do that, and it is very important that people who want to use that one, it should be very easy for them to retrieve. So we have to generate some of the applications, front-end applications, which are attractive, easy to use. So these are retrieval systems. Then presentations. How do I present the data? How do I present the information? This is why it's information system, not data system. And then the other part of the PGR informatics is analysis. Huge amount of data are lying here, like AICRP data. Okay, AICRP data is not small data, huge data. But how many people are making use of that? Very, very few. So what is the process that is required? So we require 3 sets of processes: discovery, exploration, and extraction of data. There are different sets of algorithms that are available. People who are having interest, let's say I want to make use of, let's say exploration kind of work or extraction algorithms based on my crop, my work, and all the previous and data available elsewhere, then one should be able to use them. So that is also part of PGR informatics. Of diverse information. See, initially the information is, let us say, only on Excel sheet, but it includes anything else. Anything and everything is information now, because the computer has become so powerful that facts, figures, statistics, knowledge, news, pictures, videos, anything in any language can be diverse information. And why it is so important? Everybody is, let's say, appreciative of this, because PGR informatics brings transformation in your work. On the left-hand side, let's say you have information on lakhs of accessions, their collections, there's so many things. How do you maintain? You maintain in files. Then you want to use, you want to search, you want to keep them safe, secure. So impossible, near impossible. On the other hand, the moment you digitalize them, everything is available in a system where anybody from anywhere can access. So it becomes a transformated system. Okay, it's totally transformed. So which is why the fundamental purpose of PGR informatics is not just that you people are sitting in some different places and you click and get information. Yes, that is very, very important, which is what I'm showing in this picture. That's very important, that everybody should be able to get information absolutely without any kind of competition, with equal access. Everything should be equal. But most important thing is an organized digital information system provides fair and just opportunity for all to access. If it is, let us say, in the file, somebody who is in IRI or nearby can come and access that one. People in Northeast or South or some other places, anywhere in rural, all agricultural activities are taking place in smaller and smaller places where fields are available, other facilities are there. But if you want to access information, it becomes very difficult. Now, if you can transform this one, provided that you have minimal, let us say, computational facilities, such as you have a mobile or you have a desktop and then internet, then you can get the information as quickly as easily and systematically as anybody who is also in a city or a Delhi or anywhere else. So which is why it is not just equal or fair, it is also just. That means anything which is a barrier should be removed. It is open access. Okay, you don't have to have a login or do kind of thing. No. One is equal, anybody should be able to access. Second one is fair. That means anybody should be able to access with equal facility as much as anyone else. The third one is there should be no blockade. So it is equal, fair, and just. So the systems that we develop, we ensure that we follow these principles. And why they have become important, I just mentioned, because people from NBPGR and their colleagues, when they go for exploration, collecting from different places, say 10, 20 years ago, it was fairly easier. But now not so. Farmers are aware of their rights. They know that if they're taking something from us, there must be something important in this. So they will ask you questions. They have greater awareness. There is greater awareness in the bureaucracy. There is greater awareness in the state government, state departments, in forest department. Everywhere there is a greater awareness about the importance of PGRFA and the significance attached to PGRFA and the rules and regulations perhaps governing the PGRFA. So they are becoming more and more aware. So because they are becoming more and more aware, so information must be made available as and when required, the way it is required to those people. Okay, everybody is not supposed to get information in the same way. A researcher requires a different kind of information. A policymaker requires a different. A policeman or somebody who is in the toll naka requires a different kind of information. And various international agreements coming into the force, and availability of information also very, very important. Now more and more huge amount of information is getting added every day, every hour. So they are becoming huge. So now the information is available. So it becomes our onus now that we make use of that information and develop systems that can gather that information, store that information, make it available in a systematic fashion. Fortunately, we have technologies, which is why it has changed. We have technologies to record, link, archive, do all kinds of activities. Okay. In fact, more powerful technologies are coming. We'll talk later if I get time. And they are increasing power of the computers and the network. Okay. There are three things that have happened, which has changed the way we are accessing information. Okay. People must have told you, Dr. Pratibha Brahmi must have told you the things that have changed the way we access the material, but the things are same. Means drastic things have happened. Okay, very means earth-shaking things have happened. Very, very important, significant things have happened that have changed the way we maintain data, maintain information, and the information is accessed. Okay, what are those? One is technology change. See, earlier we used to have computers or the systems that were sequential processing. That means your amount of data that you could store was limited, and the number of linkages that you could create, the layers that you could create on each data was also limited. It was difficult, not easy. So from sequential processing, like the tape recorder tape, sequential processing, to let us say digital now. Okay, where you want to select from which point you want to listen to the song, you can just start from that particular place right away. So that is random access processing. So because of that, our power has increased. The power has increased enormously. So that has helped us, that has reduced the cost, that has made things available everywhere. So we could make use of these things. So which is why the PGR informatics is one of the very, very, let's say, topmost beneficiary of this. Otherwise, it is very difficult to invest money on huge amount of servers and other things. Second one is policy change. Earlier, it was open access. Anything is for everybody. You publish it. That's it. You simply do something and publish it. Right now, also you and we, many people, many of us are doing the same thing. We say that, okay, this was there, so you published it. We do not know the importance of the privacy or the ownership of the person, of the place or the institution from where you have accessed the material, which is very, very important. The ownership or the proprietorship, it's not that they will stop you from using. Please remember, it is little different from that. The proprietorship of the people who have given this, donors, owners, okay, on the material, germplasm, is as good or as much as a patent you can develop on a material, its use, a product. Okay, so that's also very, very important. So we have to respect that. So from open access, things have come to regulated access. So accordingly, we have to plan. How do we ensure our information systems do not infringe upon their rights, but also do not take away your right to get good data, okay, correct data. So we have to manage both of them. So this policy change has changed the way we manage our databases. Third one is objective change. Initially, when people started building databases, the purpose, everything was for conservation. We are losing germplasm, so we have to conserve. So the final objective of all the activities was conservation, including PGR information management. But now it is utilization. So the utilization means I have to convert them into such tablets, such modules, so that different people who want to use them will be able to get it and digest, and then it becomes efficiently used. So for the utilizing people, they should be made available. So this is how from conservation to utilization, our focus has changed. So who are all important PGR information managers in the world? For all the CG gene banks. So when I say CG, CGIAR, Consultative Group on International Agriculture Research, CGIAR gene banks, I'm sure somebody has already told you about that. All of you know about that. CG gene banks data, there is a portal called Genesis. This is the one, Genesis PGR. So Genesis PGR, you will be able to get information of all the CG gene banks in one place. You also can get the same information on their own websites, their own databases, their own systems, but you can get them here in one place on Genesis. So we also have our own portal. They have a portal for all the CG gene banks. We have not shared our data. Our data is public access, public domain. Our data is in public domain, but we have not shared our data formally with the Genesis people, so they cannot reflect our data on their databases. Okay, but anybody else can see and use our data. Nobody is stopping. So we have our own portal. I'll not go into the story how we did that one, but please just remember that whatever I'm going to show you, or you will be able to use sometime later, we have a practical session perhaps. Everything A to Z has been developed by us in-house with hardly any expertise to start with. We did not know anything, how to do that one, how do we manage, what kind of infrastructure is required, how do we ensure that there are let's say security issues or security is not breached. We did not know many of these things, but it is now more than 10 years. Yeah, 10 years. We started our PGR portal in 2012. So it is 10 years now. It is running fantastically, so absolutely no problem. Okay, this is very important. Things do not happen overnight. Okay, we have to have perseverance. We all know about that. So how did we have perseverance? It's not easy. You will get frustrated sometimes. Sometimes funds don't come. Sometimes your algorithms don't work. Sometimes your partners do not share with the data in the form in which they should share, or timely sharing is not there. You have an experiment, and then the full data is only when you get all the 10 partners share their data. If one is not sharing, then this becomes incomplete. So when that happens, you will get frustrated. So all these things happen over a period of time. But yes, everybody has, let's say, participated in this process. Please remember, this is a participatory process. What I am showing is only something which we have developed here. What is the contents of that one? What is inside that is because of all the partners. Some of you are also perhaps partners. And at the end of this, I would like to say, end of this presentation, I'll show how you can become proactive partners with us. So in 1997, we started computerization of the gene bank ID allotment. Okay, please remember, IC number. IC is indigenous collection. IC number is not NVPGR number. So when you take the material, don't change that one and to use your institute number or something. Nothing can replace IC number. IC number is the national identity. It's India's identity. Don't change that one. For your experimental purpose, you give some codes and all different. No, no problem. Nobody should change the IC number. If something happens down the line, nobody will be able to protect you. Okay, somebody takes away. If you use IC number, the records are there here. It is considered as the national identity by all the international organizations. So your work is always protected. Your intellectual property is always protected. So in your papers, in your experiments, in your programs, everywhere, in your pedigree, If you are using any IC in your work, you should continue to use that IC number very, very properly, accurately, not changing anything. Okay. We started with the IC number, and then slowly we built databases. Initial part was building databases. And then in 2012, we launched the PGR portal. And then we started working upon that. After that, as I just mentioned, one is about the databases, that is called as the back end, that you don't see. One is about building the front end, the applications, apps, web apps. So that from 2014, we started working greatly on developing apps, web applications, mobile applications on various purposes, on various issues, various parts of the operations, like gene bank, crop oil relatives, and then registration, characterization, cryo. All gene banks have different apps, all activities. Okay. And then we have herbarium, which is e-herbarium is there. If it is not shown already, somebody will show you perhaps. We have a PGR map, we have a PGR CLIM application, we have a field gene bank. Some application is called as a G2G, gene bank to gen bank. We have a quarantine and genomic resource, just last year application. Okay. We have a number of applications that have been developed and work is going on. So some of these you can see here, these are the apps, gene bank, total gene bank, and then specific gene banks, and then germplasm registration. Perhaps some of you already know about this. The germplasm registration process is completely online. So that is developed in this as a part of informatics project. Okay. So all these things are there. We have a gap analysis, different ways of working on gaps, and then various things are there. Okay. I don't know whether I'll get some time to show that one, or in practicals they'll show perhaps some of these, like PGR map is there, gene bank is there. They're all available on your Android mobiles easily. You can download and install. So you take PGR map, open PGR map app anywhere you roam around in India. Open your internet, open your location, switch on your location, and then it will show you all those things that are being explored around you, where you are standing. If you touch that one, if you touch that one, then you will get all the details from passport to any details that are available. And then if you want to select some species and want to know where all they have been explored, they have been collected from India, you will get that data. So they are all very useful. There are many other useful utility applications are also there. See, some of them are very specific applications with the purpose that we want to show you. Some of them have been developed for the utility, for all of you to make use of that. You have some information regarding an accession, you want to know exactly IC number, you want to know more details about that. So you can get that information from some of these applications that we have. I'll show you, provided we have got some time. Okay, so these are all the good things that we have done so far, but the challenges are continuing, and they will continue. Data quality is a huge challenge, because data quality is not in our hands. You are the people who are going to generate data. Even if out of 100, 10%, 10 data points are wrong, inaccurate, okay, substandard, they will spoil the entire whole lot. The data quality is very, very important. So when you are recording data in the field, and when you are converting that one into an Excel sheet or the ways, and then sharing it with different your partners, please ensure that everything is very accurate. That means your decimal, so the way you record, the way it is recorded. Then how do we link, dynamic linking and updating, interoperability? How do we, if you want to share with different other systems, how do we share that one? For example, I'll give you practical examples, couple of examples. Number one, taxonomists have a habit of changing the scientific names. What is the scientific name of, let's say, bajra in the year of millets? Let us find out. Anybody? What is the scientific name of bajra? Bataiye. Let us know. Switch on your microphone. What is the scientific name of bajra?
Penicillium glucum.
Okay. Anybody else?
Penicillium glucum.
Correct. Okay. Anybody else? Anyone else who differs from this? No?
Penicillium americanum.
Okay. Any other? So what you have to do is
Penicillium typhoidis.
Penicetum typhoid. So everybody penicetum. So just go back and see that one. So it has been changed to not yesterday. It is now Cenchrus. Okay. So it is Cenchrus. Go back and check with the USDB taxonomy or other taxonomy databases wherever these final names are there. These are there in our textbooks and we continue to use them, Penicetum, but it is changed to Cenchrus. So when are these things happen? So these things keep on happening. They keep that when they merge two subgroups, subspecies, such things keep on happening. So we have to update ourselves. Then we have to decide whether we update right away or we update in the back end and in the front end slowly will update. Otherwise, you people will not be able to search because you will start searching for Penicetum and then you will get 0. If we convert everything into Cenchrus. Then let's say in India, they divide states into 2 states, districts into 3 districts. So these areas have to be updated. So things have to be changed regularly. It's not an easy job. Okay. One of the very, very important or the significant thing which is still challenging us is the providing information in vernacular languages, local languages. Right now it is only in English, okay? Because algorithms are in English, final data are delivered to us are in English. So how do we ensure that people who can search, those who are not scientists, but others, local people who want to search and would like to see whether their material is here. So what are their rights? How can they access? How can they ensure that if something is being used for varietal development or something else? So they want to see. Right now it is not possible. Unfortunately, we are very unhappy about that. We are trying to see how we can provide information services in multiple languages, working towards that. Okay. So I just want to thank. I earlier also mentioned that it is not one person's or one group's work. All past and present NBPG are colleagues for primary data, various kinds of primary data. You will not know that one, how deep and differentiated data they are, and their support and help. Okay, the funding from ICR in the form of national fellowship, and then various other projects with which we have purchased the infrastructure. We have people to work with us. Lots of hand-holding by those people, different global projects, etc. Now the last point. So we have where we are right now, but how you can become a proactive partner with us. Right now you have seen. Otherwise, I will show just a page and then show it to you from where you will be able to access these applications, depending upon how Ravi tells me how much time is left. But how you can become a partner with us, a proactive partner. One is a sleeping partner. We have developed something, you can just come and see and then go home. That's it. That's one thing. The other thing is you can become a partner with us. You can become a serious partner with us, with or without a project, because you have information, you have expertise, so you can contribute. And everybody will get credits for each. Please remember, a database is a very funny thing. Unlike a research paper, something is added in the database, let's say provided by you. In the beginning, everybody says, no, it is we have provided, so we have done exploration, we have collected, or we have done, let's say, characterization, evaluation, biochemical evaluation. So everybody should be credited with that. Yes, we give credit. Everybody's names will be there, are there. So you can search by your names also in our characterization database. All these things are there. In our exploration also, you can search by names. So if you your name and if you have explored and provided to our gene bank, all those things will be listed. But the debit is also yours. The responsibility is also yours. If there are something which is wrong, in future something comes up around that, so you will be the responsible. You will have responsibility for that also. Okay. So credit will certainly be given along with the responsibility. So it has to be accurate all the time. Okay, coming to this slide now. We do not have a database of farmer, conservers, and custodians. Please remember, what we have in our gene bank is a minuscule part of the total plant genetic resources that we have in our country. A small thing, a representative. In fact, a very, very good representative. But we have many other things in finished and semi-finished kind of products, in unfinished kinds of products. Okay, in the chain of evolution, it's going on. Many other things are in various places. Where are they? They are present with the custodians, they are present with farmers, conservers, they are present with, let's say, cultivated and forest, where they meet. It's usually there, you get lots of wild relatives, crop wild relatives, and many other things. So we have those. And we have traditional knowledge. So all these things are present in different areas in India, and they are present in local language. Traditional knowledge is present in local language. The farmer conservers have given a land race or a standard, the farmer's cultivar name in local language. So that name itself will have a lots of information. Name itself will carry a lots of information. So all of you can become partner with us by providing us this kind of information. At least, look, we have in our area, based on my exploration or based on my village or my place or my workplace, wherever we have this list and we have this information. If you want, you can add that one. So we will check that one and then we keep on adding that one to this data. So our 2023 work is on the first bullet, farmer conservation and custodians. We are in the process of developing this kind of database, and this is a expanding database. We are trying to develop a mobile app in which you can right away, then and there, wherever you are take photographs and that add information. So keep on increasing. That kind of things we are trying to develop. So I request your active participation in that. Ravi, how much time do I have?
Sir, still 30 minutes, 25 minutes are there, sir.
Oh, excellent. So I have lots of time.
Yes.
Now what I'll do is I will stop sharing this one.
Yes, sir.
And open. Share. So is this visible to everybody? This is the home page of, this is the home page of the PGR informatics program in NVPGR. There are different ways to reach this place. Can you see the URL, the address of this one, pgrinformatics.nvpgr.arnet.in. So you may like to note down that one, pgrinformatics.nvpgr.arnet.in. So here on the left side on the menu, you have web apps. Here, second one, web apps. When I click web apps, so you will have all the web apps listed here. Okay. So all these web apps are there. I'll just show you quickly one by one. But before that, what I would like to tell you about is these are not just developed and kept to ourselves. All these are formally launched. All these apps are formally launched. In fact, majority of them, 1 or 2 perhaps we'll be doing next year, but majority of them have been formally launched by very famous people, national or international famous people. See, our minister launched PGR map. PGR Clim was launched by Paul Gebs from California University. DDG launched a couple of apps. Dr. Sonti, who was earlier with CCMB, then with the NIPGR, and now he is the director of ICGB. Dr. Sonti, he launched one app, eHerbarium. So like this, all apps are formally launched by DG, past and present DGs, earlier DGs. Okay, so they're all formally launched. They're all can be used and quoted. Why I'm saying this one is you can take the information from here. Any information from any of these apps is considered as a good source, proper source. So all you have to do is to take information, use data, and then give in your reference. So this data accessed from this whatever www.pgr portal or GeneBank, wherever, and accessed on that date. You have to just give the data. That's it. No more credit is required to us. All of you can use this information freely. When I say freely, with freedom, full freedom. So that's the meaning, but full responsibility. So one which is a PGR portal. I don't know, in my PPT, I did not show you about where it is accessed. I think I forgot to put that one. Okay, please remember that this particular. You are seeing my PGR portal page, Ravi?
Yes, sir. Yes, sir.
Okay. So this is the PGR portal is our major app where most of the information comes and is available. So from here you can search for passport. Okay. I'm not going to show you quickly, details in that one. So it is like this. If you say plant and crop name, let us say if I say rice, so the moment I use RIC, so you will get rice or rice bean. You select rice. Just wait for 1 second or 2 seconds. So it will take rice and it will go back. Now it has worked with our server, our databases, and it has come back. Now what it will show you is, you can see here some pie chart kind of thing on the right side. On the right side, the first pie chart, if you click here, it will show you something. So what is there? IC we have 90,000, more than 90,000 accessions. EC we have 4,300 accessions. So these many accessions are there in our gene bank, of whose information we have validated. In fact, more accessions are there. Our data will have fewer, less, means some discount is there because of the lack of data validation. The moment keep on, this is an ongoing process. Every day we keep on doing validation, then this is added. So you can click on this one. So for example, you can see here on the left side, scientific name. There is a drop down here. So how many species of rice are there? Okay. So on the, if you click on the pie chart, you will see how many of each accessions are there in our gene bank. So without searching, before any searching, you already know this information. Similarly, from which country? I have just clicked on the on this particular pie chart, so you know exactly from what is the source. Okay, so all this information is there. What is the biological status? So such kind of details are there. So you can get a lot of information. How much information? How many core collections are there? And details about that. You develop core collections. You provide us with information. Okay. If the accessions are not there with us, you provide accessions also along with the IC numbers, EC numbers, and provide information. So along with your research paper, your core collection will be displayed here. So anybody will be able to access that. Our gene bank has started conserving nils and reels also. So those who are from genetics and plant breeding, perhaps many of you are from genetics and plant breeding. You know about near isogenic lines and then recombinant inbred lines and all. Nils and reels, all these are also maintained populations with the trait, specific traits, and a research paper, which is peer-reviewed research paper on that. So these are also conserved. So such information will also be available. Okay. So let me go back. Because of this floating thing, yes. Okay, I'll go to, forget about all these things. Let me go to PGR map directly. Okay, it will be of some interest to all of you. So everybody is able to see now. So this is a PGR map application. Same thing is available on the as a mobile app as well. You can right away, when I'm speaking just now, you can right away install on your mobiles and then tell me if it is not getting installed from your, particularly those who are having Android phones. So there are three options here on the left. One is what is around me. Second one is search the map. Third one is the search the species. Now what is around me is because I have switched on my internet, I have switched on my location. So it has directly taken me to New Delhi, where I am right now. So if you click on here, then it will take me that different crop groups. I'll increase the size of this one. Okay. Different crop groups. Okay. And then you click on that one. Then you know exactly which are all the crops. So you click on that one, you know exactly which are all the IC numbers. Okay. So if it is there in our gene bank, it will show you green and thumbs up. If it is not there in the gene bank, it will show you red and thumbs down. So that means you know that, okay, even if something has been, this PGR map is specifically not for looking at and indenting, asking for the material. No, it will help you in knowing exactly from where, what kind of things have been collected. See, when you want to, whether your objective is for exploration and collection, or whether your objective is to study the occurrence, distribution of a particular species, crop species, any kind of things. For example, developmental agencies want to know, means forest departments want to know, for example, crop wild relatives, etc. So things are there where they are there. Okay, some kind of Ramsar, wetland development, things are going on in Tamil Nadu. Various places are there. In Kerala, they wanted information on few other things. So you will get information because this has been accumulated over a period of time. Experts go there, have collected. Whether it has gone to gene bank or not is a different question. That's altogether a different issue. But people have gone there, identified, collected. That means it is a place of occurrence. So this will give you a very good surrogate, very good stepney for distribution occurrence. So like that, you can see this one. And then search the map. One is what's around me. Then search the map. Search the map is you just go anywhere. Any place. You can click any place and then. So what I'm going to do is I'm going to click on a very remote place. Andaman. So then I'll say that search here. So it will give me cereals and tubers and all the things that have been collected from there. Okay, maize, rice, mainly rice has been collected. So how many rice have been collected, which year, all the details, what is the name, if you know anything. So how many have reached our gene bank and how many have not reached our gene bank. So if you are part of any future exploration programs, you know exactly by this, okay, these are the places where people collected, but have not reached our gene bank. So if we find out, we can collect again. Not a problem. The third one is about search for species. So you can search here by, let's say, scientific name. I will use sorghum. Okay, no. Okay, because I'm taking an example of a millet, I'll use Alucine. So you can see here. So all these dots are not dead dots. They are not something which is just for color purpose. Each one of them is interactive. You click on that, you will click on this, and then you get passport details. Okay. So you just name any crop like that. Search for species. I'll search for any one of you. Come up with your own species. Don't give rice, wheat, maize kind of things.
Chrysanthemum.
Challenge.
Chrysanthemum modifolium.
Huh?
Chrysanthemum modifolium.
Chrysanthemum.
Yes.
CHRY. Chrysanthemum. Which one?
Modifolium.
Mori.
Modifolium. Yes.
Yeah, it is there here. So let me see. So these are the places, these are the ones.
Okay, thank you. Right, thank you.
Morus levigata.
Yeah, 1 second. मैं एक करके pass हो जाऊंगा ना. I'll just pass one test. Let me see first whether they are there in our gene bank or whether it is there in, because my question then will be little different. I'll explain to you why I'm doing this one. So whenever we are seeing a species which usually is not conserved in seed gene bank, okay, so you will get all red thumbs down because they will be there somewhere else. So if you are asking those questions, then you are also part of the same team. Then you will become our partner quickly.
Yes.
Tell us where these are perhaps conserved, which are the institutions, which are the KVKs, Ataris, institutions, private places, anywhere where these could be possibly available. So we will contact all of them and find out if these IC numbers are with them. Then we will update the data. It will go to FGB, Field Gene Bank database. Okay. Okay. अब बताओ भाई Morus, another
Morus levigata.
Okay, another field one. Levigata. Yeah, it is here.
Yes.
Okay? So these are the places where they have been collected. But I'll show you now. So you will get again thumbs down. Why thumbs down? Because either they are there with Hosur or some other place. I don't know where else they are there. These Morus germplasm are maintained, but they are not with us, right?
Sir, field gene banks, sir, they are maintaining that.
They are maintained in field chain bank, and a lot of it is also available in our cryo. Our
Yes,
cryo also they are there. That's it. We are rationalizing all these. But if you people, which is why I'm encouraging you to ask this question. You are asking this question that you found out now that yes, the information is there up to from where they have been collected, but information is not there where they are conserved, where they are available. So this is as good as useless, right? So it has to be completed. So we should know where they are conserved, where are they available. Because what happens when they were collected, let us say, which is the year, we don't have year here, which once they collected 573, this is a recent one only. When they were collected, somebody else would be there. Now next generation person will be there in that field gene bank place. So the information may be lost, may be incomplete. So that should not happen. Once it is digitally complete, anybody will be able to maintain and access everything. Means they'll become useful for all of us.
And that kind of how to update, sir, from our side, how to operate, because Mulberry concern, CSARC Central Silkwood is a nodal center and NAC site, and how to update that information. And already we send, our office send one letter for this information.
Yes, they are there. I think you are also trying to share the characterization data with us.
Yes.
So you are the person. Okay, excellent. So
Yes, sir.
Okay, good. We are trying to do that one. See, it's like this. You all know about descriptors and descriptor states.
Yes.
So the crops that we are managing, we already have standard descriptors and descriptor states. These descriptors and descriptor states are officially published, officially known ones. Means anybody doing experiment will not be doing anything. They must be using these standard descriptors and standard descriptor states. Like flower color, you cannot say that this is dark reddish maroon kind of thing. So you cannot give this kind of description, right? So there are different states, so people will give that one. So wherever the new crops are come with us, so we start setting up a process where our descriptors we check with that one. Because what will happen, you or somebody else in different crops, they use dust descriptors, PPUFRA, but they don't match with our descriptors. Please remember, dust descriptors are for different purpose. Our descriptors were more of a botanical and taxonomical importance let's say provided or is given to those descriptors which are being used. They appear very normal and useless sometimes. Ligule color, ligule size, etc. Isn't it? But they're all very important. They are very important in grasses. So this is how it is. So this is in process. You don't have to update on this database. We update on that data. Right? One point we update that one.
Okay.
Excellent.
Okay, thank you.
But one more point is, let us say you have your own database where you have pictures of various things. The fruit pictures are there. Many other things are there. Your biochemical information is there. So we can link those things also on our application. So it becomes easy for someone. In one place they come. From here they go to all other places. So we are trying to do that. We are trying to.
Yes, sir. We want to such a way only, sir. But that's why I am asking how we link to this NPPGR link.
We will do that.
That's why we send a letter.
Which is I already mentioned that we have received. So we are in the process. We are building a second version of PGR portal, which is fairly easier and then user-oriented kind of one. When in 2012 we were not that brave enough. We were only learning at that time. So now we know we have 10 years of experience. We are going to launch a very different kind of application, very easy to use kind of one, modern style. So in which we are going to give links to many other things. Let's say recently there is a online. It's not an app. It's an online information published by all the tree species that are available in Northeast. Their pictures, location, etc. That's very important. Anywhere, let's say traditional data, medicinal data, everything is important for us. So we are going to link most of these things to ourselves. So we will do that one. Hopefully, it will be done in this year only.
Okay.
Excellent. Now, what
Okay, thank you.
thank you. Ravi, somebody else will show NHCP, I hope. What I'll do is I'll show characterization, which is characterization is important. See here, characterization. All of you, please give your attention. This is characterization and evaluation database, although we have only characterization data right now. So you can see here, 2,27,957 accession data are there. That doesn't mean that 2,27,000 unique ones are there. Please remember, we keep on adding data of the same accession done 10 times. We don't mind at all, because if it is done somewhere else, in some different season, different year, all those things should be available to the next future users, because they should be able to compare all of them and then see, is this something really highly inherited ones are there or somewhere, because there are some which are not 100% characterization data, such as different metric data are there. Yield related data kinds of things are there. So they differ from place to place, season to season. So we keep all the data. But crop wise, you will get different data. So how many indigenous, exotic, how many species data are there? See, 211 species characterization data are there. There is no other place you will get this kind of information. Although you don't expect that in your own species you will have information on all the accessions. That is going to be your own responsibility to generate that data and share here. Okay, and then become owner of that one. I'll show you that one. There is search data, no, advanced free search data. So if you have any one of you has provided characterization data, apna naam bataiye. Please let me have your name so that I'll show you right away. Anyone has shared your characterization data with us? No? Then I will just add our colleague who is not with us anymore. See, I put that name and then all the names are here, which year, where they have done. So if you carry out information, if you carry out the characterization and provide us with the data, your names also will be there. This is sufficient of an evidence for all your interviews, what else, promotion kind of things. This is sufficient evidence that you have done this one. Okay, it's very important. Please remember, ICR agrees, ICR accepts this as proper evidence that you have done and it is available with us like this. And then by crop also you can search by just crop, and you can search here whether the data are available or not. So all these things are there. There is a advanced search data is there, which I'm not going to show you that one. In a specific crop, just select the crop and automatically its descriptors will be loaded on the back end. So you can one by one, you can select which descriptor and then you can say minimum, maximum, etc., and then submit and you will get the shorter list. So all those things will be there. There is a graphical view representation is there. Okay, I'll just say barley, take anyone, 75% emergence, submit. So you'll get this kind of data quickly. See, isn't it fast enough? I'm just selecting and the graph is quickly generated. I'm sitting at my home, I'm not in office. So that means it is working pretty fast. So we have standardized our algorithms to provide you data with very fast. So you know exactly which range, how many accessions are there. Accordingly, we'll start select shortlist and then demand for these accessions. So similarly, many other things are there. I'm not going to show that one right now. Somebody is going to talk about journalism registration. Ravi, is there
yes sir, Dr. Veena Gupta.
So
yes sir.
Wednesday class is there, sir. Wednesday one lecture is there, sir.
Okay.
Wednesday Veena madam will give, sir.
Excellent. Anji, somebody said hello. Anybody has said hello? Koi baat nahi? Chaliye. Okay, so this is the page where you will get all the data, or you can say, how do you go from our NVPGR website? I'll show you that one. PGR databases on the top here. PGR databases. Click on PGR databases, so you will get a number of these. So you can click them and you will get, and many other applications are there. Okay, so this is all. But the take-home message is messages. There are 3 take-home messages from my side. Number 1, whatever information that has been generated by NBPGR on its own or with the partners or partners on their own, and they have shared data. All these data which are in proper format are available in our information systems. They are there, number 1. Number 2, all these information are available as open access. Anybody, did you see anywhere I entered a user ID password? No, no user ID password. They are available as open access. All of you can make use of that one. Number 3, all of you are becoming very, very important partners for us. If not, you are already, you are going to become very important partners with us, especially for perennial crops. Because perennial crops, we do not have information on their location where they are conserved. We also want to have information on their characterization and trade specific evaluation or trade data. The moment we have that one, you will become owner of that data, because it is already, the moment things are in public domain, that okay, this is available there, you are already owner of that one, because they are with you physically. Information is in your name, so you become owner. More use will be there. You will also become owner. If it is, let us say, industry wants to make use of your shahtooth, that means your mulberry, or any other industry, medicinally important, pharmaceutically important, they will contact you. It is there here. You can sign MOU based on this information. So it becomes very, very important. Everybody is going to win by sharing more and more data. Okay, thank you very much.
Anyone have doubts? Sir, our next speaker is also joined.
Okay.
Yes.
So I'll say thank you to all of you and then exit.
Yes, sir. Sir, thank you very much. Thank you very much for your valuable presentation and enlighten the all activities of PGR Informatics to the participants. And sir, thank you, sir. Thank you. Thank you.
Thank you very much. I also I want to thank Vijay Kumar Mandal, who is sitting there.
Yes.
Here, he is participating in this training program more than all of you. He's attending each and every lecture.
Yes, yes, yes.
In another 1 or 2 years, he will deliver a lecture.
Yes, sir. Yes, sir. He is more than our more than a speaker. He is attending, sir. Yes.
Thank you very much. Bye bye.
Thank you, sir. Thank you. विजय मंडल जी, हमारा दूसरा स्पीकर भी and our next speaker is also online, and he joined already in meeting. Now I am. It is a time to sir, Lalji Singh sir.
Yes. Hello.
Sir. Sir.
Good afternoon.
Sir. Good afternoon, sir. Good afternoon, sir. And let me introduce our next speaker. He is nothing but Dr. Lalji Singh. He is a Joint Director, Scientist E and Head, Botanical Survey of India, Andaman Nicobar Regional Circle, that is a port player. And his nature of work and achievements are, he is expertise in revisionary, exploratory, and conservation studies on the flora of India, especially in Andaman and Nicobar Islands. And he worked more than 1 decade in Andaman and Nicobar Islands, and mostly he focuses on plant morphology, anatomy, especially aerial roots and dicots. And he described more than 17 taxa, including a new genus to science and various additions to Indian flora also. And he mostly worked on the taxonomic revisions and systematic studies on the taxa of Cycadaceae, Loranthaceae, Musaceae, and Gingiveraceae. And he is the first time and the first person he described the phenology of 73 species of Andaman and Nicobar Islands. And he is a good teacher, and he guided a lot of students on a honorary basis. His students also given the name of his new species, that is Dendrophytae laljae. It is a Loranthaceae member, and Portulaceae laljae and Cyrostria laljae. It is a Vinegaraceae rubiaceae member, and he is a fellow of Linnean Society and fellow of Botanical Society and fellow of other different societies and fellow of TOSI published more than 135 research papers, and he personally established and invented some reusable eco-friendly drinking straws also. And sir, please, sir, kindly share your expertise and knowledge to our participants. Here, sir, the participants are from different organizations, and ICR and non-ICR organizations also there. And sir, I am welcome you. Please kindly continue your presentation, sir.
Thank you so much for inviting me.
Yes, sir. Sir, share your share screen, sir, your presentation. Sir, presentation mode, sir.
Hello.
Yes, sir.
Slide.
Yes, sir. Yes, sir. Sir, please continue, sir.
Okay.
It is visible, sir. Alright, sir. Yes.
Okay. So, namaskar to all of you and good evening, distinguished dignitaries, invitees, and dear participants. It gives me immense pleasure to be part of this training program on management and genetic resources. I am thankful to the director, Dr. Gyanendra Pratap Singh Ji, and Dr. R.K. Gautam, Principal Scientist and Head Division of Germplasm Evaluation, and members of organizers of this event for inviting me and giving me this opportunity to share with you my views on plant diversity of Andaman and Nicobar Islands and overview with special references to the agricultural potentials. Being a botanist, it's my great honor and privilege to be a part of this event as a speaker. Our country has well recognized for its rich and unique plant diversity since immemorial time. The history of botanical exploration in India dates back to the entry of Portuguese for trade of plants and other forest products. Subsequently, Arab, Turkish, Dutch, French, finally Britishers trades entered in our country. Britishers started East India Company and subsequently established garden at Shivpur, Calcutta in 1787, following the recommendation of the Colonel Robert Kidd. British Government of India in 1890 established the Botanical Survey of India under the direction of Sir King George. He was the superintendent of the Royal Botanic Garden as its first ex officio director of this premier organization of the taxonomy of country. The organization's mandate was explore, collect, identify and document the rich plant resources of the erstwhile British India. So this is a image of the Sir George King, first ex officio director. After independence, for broadening the knowledge base on plant resources, it was recognized in 1954 and Dr. E.K. Janki Amal was appointed as an officer on special duty by Government of India, a scientist who was born in the southern Indian state of Kerala in the 19th century study a wide range of flowering plants, and rework the scientific classification of several families of the plants. Subsequently, a number of regional centers were established in the various phytogeographical region of country, and as on today, BSI has 11 regional centers, 4 units and 2 botanic gardens. Now, AJC boasts Indian Botanic Garden, Havda, and Botanic Garden of the Indian Republic at Noida, where working with prime objectives such as exploration, inventorizations, and documentation of phytodiversity, with more emphasis on protected areas, hotspots, fragile ecosystem, and publication of national flora along with the conservation strategy. BSI maintains more than 3 million herbarium specimens, including 1888 types, and thus stands as a national repository of plant resources. So this is a glimpse of the Andaman Nicobar Regional Center after independence, broadening of the exploration. In 1972, Regional Center of BSI established in the Andaman and Nicobar Islands. This is a glimpse of our regional center in the islands. We have a national repository, herbarium, acronym is PBL. Till date, we have made 255 exploration tours and 40,000 herbarium specimens, including 422 type specimens, and discovered 3 genera and approximate 116 new species as new to science by the scientists of this regional centers. So this is a glimpse of our national repository herbarium having 40,000 already I told, and 419 type specimens, approximate 400-600 specimens belongs to the lichens. And we have a virtual herbarium with metadata is available on the website of BSI, and this is one of the oldest herbarium specimen deposited in our PBL herbarium Andaman, collected in 1891 by Dr. Dufresne. We also have a museum, approximate 500 exhibits for the public awareness, and there are some specific and curiosity specimens are there. Largest infructescence fruit axis over 4 meter long of the endemic banana species Musa indenmanensis, which was discovered in 2014. This is another glimpse of our museum. So right side, over 4 meter long fruit axis of the Musa indenmanensis, and another is interesting, longest pod among the climber birds. This is a glimpse of the anatomical details of the roots and phloem wedges, first time discovered by myself in the some Vignonaceous genera, displayed in our museum for the public. And we also maintaining the garden about 16 kilometers away from the Port Blair, and approximate 600 species are under conservation. So this is a glimpse of our garden. We have various sections: medicinal plants, aquatic plants, Navgraha Vatica, succulent plant section, wild gingiber, and Musa plants. And this is a glimpse of the bamboo situm. And we also have a succulent plant section. Approximate 11 species are under ex situ conservation in our garden. However, in our country, the plant conservation was deeply rooted in the culture and tradition from the ancient times, and preserved the plants for the various regions. Older generation preserved for the mythological regions, whereas the conservation ethics of the cost have changed drastically with the time due to the biotic pressure like intense human activities, dependency on plants for their survival, and other developmental activities. The rich habitats are gradually depleting day by day. And not only in India, all over the world now it is a very sensitive issue how to conserve the plant diversity. And the anthropogenic pressure and development activity different parts of the world has led to the increasing need of using the land for the human habitations and agriculture. This has led to the reduce the habitat of phytodiversity. So every year, more than 2,000 visitors visited in our garden, students, researchers, trainees, and various society members in our garden. So floristic diversity of Andaman Nicobar Islands is a very rich and unique in our country. India is noteworthy for the biodiversity with 2.2% of world's land areas having 11.4% of flowering plants. Plant diversity of India stands at 54,733 taxa, including 21,849 angiosperm, 82 gymnosperm, 1,310 pteridophytes, 279 bryophytes, 2,961 lichens, 15,504 fungi, 8,979 algae, and 1,257 microbes. This record is based on our published Plant Discovery 2020. In India, the floral diversity is concentrated in 4 phytogeographically unique regions: Himalayas region, Western Ghats, Northeast India, and Andaman and Nicobar Islands. So, Andaman and Nicobar Islands possesses distinct identity, not only because of geography, history, culture, but also because of the unique and rich biodiversity of natural ecosystem, consists of tropical evergreen forest, wetlands, mangroves, and coral reefs. It is also one of the hotspot of the biodiversity. It is well known of the history. On December 30, 1943, Netaji Subhash Chandra Bose hosted first independent Indian flag in Andaman Nicobar Islands at Gymkhana Ground, now named at Netaji Stadium at Port Blair, Andaman. And this place is also announced the islands the first Indian territory freedom from the British rule. So this is a summary of the geography of the islands, and we have 9 national parks, 1 biosphere reserves, and 93 wildlife sanctuary. Total islands approximate 572, inhabited islands in 31, and highest point of the peak in group of Andaman Islands is Saddle Peak, and highest point in Nicobar is Mount Thulier. This is a glimpse of the highest point in group of the Andaman Islands, Saddle Peak, approximate 732 meter high. And this is a glimpse of the Mount Thulier, highest peak in the Nicobar group of islands. And this is a state tree, Pterocarpal Durval Joadis, commonly called as pride of Andaman. And Lagestomia hypoleuca is a state flower of the Andaman and Nicobar Islands. And Andaman Nicobar Islands having 2 types of forest, sorry, 3 types of forest, and littoral forest comprises mangrove forest, beach vegetation, tidal and swamp forest. Inland is evergreen forest, deciduous forest, grassland, and hydrophytic forest. and same composition is recorded from the Nicobar group of islands. So this is a glimpse of the mangrove vegetations. This is a tidal or swampy forest, and you can see a beautiful glimpse of the nematophores. And this is a glimpse of the beach of the Nicobar Islands. And here exploration is very tough. Some geographical status of the islands are very tough and difficult to reach inside and explore the plants. So we also have a waterfall, natural waterfall, and this is a glimpse of the waterfall situated in the Little Andamans. And we have a very rich green grassland, Muru, photograph taken from the point of the Muru. So floristic diversity of Andaman Nicobar Islands dates back to 1791, when Colonel Kidd visited the islands and introduced some Andaman plants, the Royal Botanic Gardens, Calcutta, which were later described in Roxburgh Flora of Indica, 1815. Thereafter, in 1839, Helfer, Russian geologist, visited the islands with a view to ascertaining mineral resources and also made extensive botanical collections. But these, after his murder by Aborigines on the North Andaman, were unfortunately mixed with his collection tangerine plants, and all were labeled as a tangerine and Andaman, which resulted in a great deal of confusion. Subsequently, botanists like Purge, Parrish, Prain, King, Rogers made some botanical studies between 1866 and 1903, which together and with his own efforts established Parkinson 1923 to bring out a flora of Andaman and Nicobar Islands. Thereafter, flora of Andaman and Nicobar Islands has been explored and recorded time to time by various workers. This is one of the glimpse scientists of BSI approaching for the exploration, plant exploration. And this is another glimpse, myself and teams of the BSI along with the forest visitors, forest officials approaching for the plant explorations. This is a field exploration, glimpse of the field exploration and plant exploration by BSI scientist and researcher along with the merit researcher of Kew Botanic Garden, UK. And this is a more recently documentation of the plant diversity of Andaman and Nicobar Islands. So this is a current status of the plant genetic diversity. Approximate 2,433 angiospermic flora have been recorded from these islands. Gymnosperms 9, pteridophytes 145, bryophytes 76, lichens 565, algae 182. So total floristic composition is 3,410. This is a current status of the flora of the plant diversity. But here gymnosperms are very poorly represented, and Cycas with 3 species, Netum with 4 species, Nagia and Podocarpus with single species. And dominant family like other geographical region of the country is Poaceae. Poaceae is top with the 197 and Leguminosae followed by Leguminosae 172. So endemism approximate estimated 10% of the total flora in our islands. So the most interesting feature of the flora of Andaman Nicobar Islands is discovery of 5 endemic genera: Nicobariodendron, Pseudodiplospora, Pubistylis, and Spironthera, and more recently, Septomeranthus hemiparasitic genus have been discovered by myself, and it is more interesting, all genera have been discovered from the Nicobar group of islands, not belongs to the Andaman group of islands, and approximate 315 endemic taxa in angiosperm, 2 taxa in gymnosperm, 6 taxa in pteridophytes, and 8 taxa in bryophytes, and 101 taxa in lichens are recorded as endemic to the islands. So these are the present status of the endemic taxa in the islands. Most of the endemic taxa are restricted to few and small geographical area and are the key floristic elements of these distinctive islands. They are more valuable due to narrow and restricted distributional range. In this condition, the dispersal rate of the fragmented population is not as fast as the change in the landscape. Their survival is at the risk as the species once lost will be the last forever. Also, interesting is the many economically important as well as ecologically significant resources, especially endemic plants, are vulnerable to the anthropogenic disturbances, natural calamities, and climate change. Of the different threats to the islands, phytodiversity, the climate change can be considered the major threat to the island's biodiversity. The group of cryptogams, especially algae and fungi, remain considerably required intense floristic survey that will contribute to the update knowledge of the cryptogamic flora of these islands. Continuous discovery of novelties from these islands are strongly evident that these islands are rich and unique in plant diversity. Many endemic plant species, including non-indigenous, are also known from a high medicinal value and other potential in these islands. So these are the some recently described new species from these islands. This is a glimpse of recently discovered new genus, hemiparasitic genus, Septomeranthus, and is a monotypic species is Nicobaricus. As I told, this genre discovered from the Nicobar group of the islands. This is another and more recently discovered new species, belongs to the Rubiaceae coffee family, Rubiaceae, and it's my great honor and privilege to share about this species. This species named after my name and represent the first record. And this is the first recorded from the Nicobar group of islands. Dendrophyll algae belongs to the Laurenthaceae family and having all potential of the medicinal property. And this is a glimpse of the pteridophytic habitats. The pteridophytes are quite considerable in these islands with approximate 6 endemic taxa. However, the knowledge of other lower groups, especially bryophytes, algae, fungi, is still insufficient and considerably requires intensive floristic survey that will contribute to update the knowledge of the pteridophytic flora and other cryptogamic flora. So this is one of the endemic species, but nowadays anthropogenic pressure are there and this is the major threat in that habitat. So this is one of the gymnosperm. Gymnosperm is a group of seed-producing plants that include conifers, cycads, ginkgo, and gnetophytes. Cycads are indeed the most ancient and archaic plants and have been referred as living fossils. They evolved during the Carbo-Permian times, approximate 280 to 300 million years ago, and reached to their climax in Jurassic times, coinciding with dinosaurs of animal world. This group achieved its maximum diversity in the Jurassic and Cretaceous periods when it was disturbed almost worldwide. At the end of the Cretaceous, when the non-avian dinosaurs became extinct, so did most the cycads in the North Hemisphere. So nowadays, some of the species in that groups are struggling for surviving on the Earth. Cycads habitat in Asia have suffered a severe reduction and degradation over the century, and these pressures will invariably continue and even increase in the near future. In India, the cycad flora is represented by single genus Cycas, which occurs naturally in Andaman Nicobar Islands, Andhra Pradesh, Tamil Nadu, Karnataka, Kerala, West Bengal, Bihar, Jharkhand, Odisha, and states of Northeast India. Nearly 17 species of cycas are presently known from our country, of which 3 species are found in Andaman and Nicobar Islands. So, gymnosperms are poorly represented in the islands with 3 families, Nitaceae, Cycadaceae, and Podocarpaceae. The family Cycadaceae represented by only Cycas genus with 3 species. So Cycas comprises only 3 species in these islands, and 2 species, namely Cycas dharmarajai and Cycas channi, are endemic to these islands. And this is a glimpse of the Cycas channi endemic species, characterized by 2 horn-like structure in the female sex organ, that is megasporophyll, discovered by myself from the north group of the islands. And this is another Cycas endemic species, Cycas dharmarajai, characterized by the trunk with strongly swollen, has been often branched, polymorphic aerial roots, sterile apex, megasporophyll with well-defined 10 to 28 paired lateral hook-like structures, 2 cytogenous mucilage canals in the leaflet. This is anatomically also very distinct from the other species, and this species is also discovered by myself. And this is another species, Cycas gelenica, recorded from Andaman as well as Sri Lanka. Earlier, only 1 species are known from the Andaman and Nicobar Islands. This is another genus, Netum. Netum with 3 species. This is a climbing species. And this is a tree species, Netum latifolium, glimpse of. And some of the genetic resources are available in our islands with the high potential value of medicines and other economical aspects, as humans have a long history of using the native flora for agriculture, horticulture, and medicinal purposes. Traditional healthcare practices of indigenous people of taking care of human health is considered as the base from which all other systems of medicine have originated, developed, and flourished. Medicinal plants have played an important role in primary healthcare system. Among the tribal people of the Andaman and Nicobar Islands, tribes of these islands are partially or completely dependent on forest resources for medicine, food, and fuel. The Andaman group of islands are inhabited by tribes Great Andamanese, Ongis, Santanese, Jarvas, and which are the Nigrotic star. The Nicobar group of islands are inhabited by the tribes of Nicobarese and the Sompen, belonging to the Mongoloid star. The Ranchi and Korain are the tribes community of mainland of India and Myanmar respectively. They were brought here for the timber extraction work during the British times. So several plant resources are available in the islands for genetic improvement of the agriculture, horticultural, commercial crops. The genetic resources of the islands also include the valuable timbers of the commercial importance, canes, bamboos, and others. So this is a glimpse of one of the palm species. Approximate 31 species of palms belonging to 14 genera have been recorded in these islands, of which 23 taxa under 6 genera are endemic, and rattans with 18 taxa under 3 genera, of which 11 taxa and 3 under 3 genera are endemic, have been recorded from these islands. So this is a Bentinkia nicobarica. Only 2 species of genus have been recorded, one from the Western Ghats and one this is species in our islands. So Bentinkia stems are the used by local tribes, especially Nicobaris and Swampen, in house and fence construction. This is another endemic and vulnerable palm species, Pinanga menai, and the seeds of this species are chewed as a substitute of the betel nut, and its inflorescence used as a decoration of the body, usually by tribes. So this is a glimpse of the 2 endemic terrestrial orchids. Approximate 152 orchid species have been recorded from these islands. So 2 species, one from the Nicobar and one from the Andaman group of islands, Elophia, have been recorded as an endemic species. This is a Vanilla andamanica, one of the endemic species, but nowadays its population is under threat and restricted, very limited in the locality. This is another species only recorded from 2 or 3 locality and with few individuals, Vanilla sanjapai, and this species named after our former director, Dr. M. Sanjappa, and very unique and characterized by the pubescent hairs on the leaves of the flower. So this is a glimpse of the one of the highly medicinal valued Etlengera fengeli gingivers. Gingiver is represented by 29 species in our islands, belongs to 10 genera, and part are used by the tribes. The leaf paste of this species is applied on the body during the honey collections from the honeycombs by the tribes and local peoples. So this is one of the unique species, and these species are under XC2 conservation in BSI garden. And this is another species, Gingiva pseudosporosum, more recently discovered from the North Andaman group of islands by myself, and it is characterized by the tuberous root, a small red colored pseudostom, arceolate or proliferate inflorescence, but very long underground peduncle, orange red vermilion lotus shaped dehisced fruits, and red colored seeds covered by the white aromatic plant. And plant parts are used by the tribes of Andaman and Nicobar Islands for curing the various alignments, abdominal pain, and athletic troubles. This is another endemic and threatened species of the gingiva, Bosenbergia siphonantha. So this is a unique species of banana discovered in 2014. And in Andaman and Nicobar Island, the family Musaceae is represented by single genus Musa, comprises only 4 species: Musa acuminata, Musa bulbiciana, Musa indenmanensis, Musa paramgitiana, of which last 2, Musa indenmanensis and Musa paramgitiana, are endemic to these islands. Tribes and rural peoples have a close relationship with this species, and seeds of used bright basal portion of the dark green cylindrical spathy inflorescence, and seeds are used for the various alignments, especially jaundice and blood pressure by the local people. This is a glimpse of the another endemic species of the Musa paramjitiana, and this is a Mangifera andamanica endemic to these islands. We have a very unique and peculiar species of the mango, Mangifera nicobarica, and very restricted in distribution, only found in the few locality of the Nicobar group of islands. And this is a highly valuable plant, Arophia cachalica, also used as a bee repellent. And this is another economically important plants, Uvaria nicobarica. Fruits are usable and highly medicinal valued and used by the tribes. This is another endemic species under severe threat and endangered state. We have a wild semicarpus, wild cashew nut. This is a semicarpus kurjai. Plant parts are medicinally used by the tribes and Nicobar Islands, curing of the various alignments. And we also have a endemic Dylinia species, and plant parts are used by the tribes Andaman and Nicobar Islands. Dylinia only have a two species in Andaman Nicobar Islands. This is a Carandus Carissa andamanensis. It is in taxonomically very distinct from the commonly known Carissa carandus species, and this species also very restricted in distribution and under threat. And we have a 7 species approximate of the nutmeg. This is one of the endemic species, Myristica andamanica. This is another endemic species, Nima andamanica. Flesh pericarp is medicinally used by the tribes. We also have a very good population of the Garcinia, and plants are used by the tribes and used as a fruits or medicinal purposes. So besides normal plants, hemiparasitic 4 family, Laurentheaceae, Vesicaceae, Arobuncaceae, and Cultucaceae have been recorded, from these islands, and Laurentheaceae with 9 species, out of which 4 species have been discovered by myself and co-workers from these islands, and plant parts are used by the tribes for the medicinal purposes. And this is a glimpse of another hemiparasitic species, Spirula paramjitae. This is a Ficus andamanica, endemic species of ficus, very good resource for the herbivores food. And xerophytic plants are very poorly represented in these islands. Only single species, Euphorbia epiphyloides, as a endemic recorded from these islands, but very restricted in population, few in population and restricted in the locality. Only 2 or 3 I saw in last 10 years exploration in 2 forested areas only. So we have also a very good diversity in the sea grasses, and but only few are recorded, and there is a lot of scope for the taxonomic study. So exhibit a low taxonomic diversity, and approximate 12 genera and 60 species worldwide recorded, and out of which 9 species can be seen in the Andaman and Nicobar Islands. So this is one of the glimpse of mangroves. In India, the mangrove flora is represented about 59 species under 41 genera and 29 species. The Indian mangroves covers about 4,827 square meter area, and a total 58 species under 28 families are representing in the flora of Andaman and Nicobar Islands. This is a present status of the main mangroves diversity and facultative. So total obligate mangroves is 36 species and facultative is 22. This is a one of the glimpse of Sonresia alba. This is a Nympha fruticans. This species named after the characters of the fruits. And we also have a common hydrophytic vegetations. There is a no endemic taxa, only some of the species Nymphia, Nymphoides, and other few Potamogitans, Ipomia are recorded from these islands. So besides endemic flora, many non-indigenous plants are also known for high medicinal value in these islands. So this is a glimpse of the Trichogenthis tricuspidata. Its vegetative parts and vary with fleshy pericarp are used by curing the various ailments, especially by the tribes Jarva and Ranchi peoples. And this is another Momartica cochinchensis. This is another species of the Diospyros discolor. Approximate 6 species have been recorded under this family, and all are used by the tribes. And this is a Aphanomyxis polystichia. Seeds are used in the various traditional system of medicine, and seed oil is used in the making soap and a lubricant agent. Capsule is eaten by bat and other frugivores. So this is a edible capsule and commonly called as Andaman grapes, and sold in the market and used as antioxidants by the tribes. And this is another berry fruits found in our islands and very good in the population. So this is another edible plants, Champacaria mammaliana. Leaves and edible drupes are used as vegetables and various traditional system of medicines. So this is a thousands of drupes hanging down recently on a climber. Vegetative parts and fruits are used in the various traditional system of medicine. But last 5 years, I had tried but unable to germinate these seeds. And I never seen eaten by the any wild animals in our garden or any other forest. So we also have a population of the Malotus philippines. This is a valuable source of the natural dye used by the preparing of sindoor, vermilion, kumkum, which is also used as a medicinally by the tribes of Andaman and Nicobar Islands for curing the various diseases. We also have a approximate 4 species of the Heliocarpus. This is the glimpse of the one of the Heliocarpus species. So this is a economically important fruit tree, pandanus, endemic to these islands, and this is a main source of the food for the tribes of these islands. And this is a teca, tubers of the teca used as a potato by the tribes. So this is a state tree commercially called as padak, and also it is locally known as pride of the Andaman, commercially high value for the durable timber. Its wood was extracted from Andaman forest and exported to UK during the British rule in the islands, and padak wood was largely used for the construction of Buckingham Palace by Britishers. So this is a Licula peltata, and this leaves used for the thatching by the local peoples and tribes peoples. And we also have a approximate 18 species of the cane. This is a glimpse of the Calamus andamanicus, and approximate 40 species of the bamboo are recorded from these islands. And BSI scientists have been explored the potential of the bamboo as a reusable, eco-friendly drinking straw, a novel way to replace the plastic straw. So it has been restored for the patent, and this is a glimpse of the tribes in islands plant diversity is very rich and unique. So tribes and rural peoples have a very close relationship with the nature and are dependent upon the forest products. This is a glimpse of the Jarwa tribes, and Jarwa tribes decorated their body with the plant parts, especially leaves, bark, fibers, flowers, etc. So most of the indigenous species are sparsely scattered and confined to a few localities which face the many significant threats such as land transformation, shifting cultivations, and others, leading to the habitat loss and loss of species which are only known from the type collections. Additionally, anthropogenic pressures, invasive arthropods, herbivores, and threatening the local flora. Several a number of species known only by the type collection and description of no further record for the other collections. So only known, for example, one of the example, endemic rice crop have been recorded in 1998, Oryza indandamanicus, but thereafter there is a no collection. I am also trying since last 12 years, but unable to spot out in the forested area or adjoining areas of the locality. Like that, Musa paramjitiana, also known only from the type locality, not any other localities have been spotted for that one. So this is a glimpse of the natural calamities of the on the plant diversity during tsunami. So most recently, we have reported the invasive arthropods herbivore poses a grave threat to the cycad population in these islands. So this is another threat, and once such type of species discovered, which prevent from the extinction through the multiplication in vitro as well as the introduction in the natural habitat. So this is a glimpse of the beauty of the Andaman Nicobar Islands. Andaman Nicobar Islands have a very fragile ecosystem which shows the unique characteristics of association of three types of ecosystem, especially marine, mangroves, and inland forest ecosystem. So this is a Sangam, junction of the three types of ecosystem together. So this is a beauty of the plant diversity of biodiversity of Andaman Nicobar Islands, and a great challenge lies in the protection of this unique ecosystem which safeguarding the livelihood security of people of these islands, as wild species related to the agricultural crop wild relatives can increase the adaptive capacity of agricultural system around the world. They represent a large pool of genetic diversity from which to draw new allelic variations required in breeding program. Crop wild relatives have been extremely valuable in adopting crop varieties to changing the disease pressures, farming practices, market demands, and climate conditions. Unfortunately, crop wild relatives are a threatened resources and measures need to be taken to protect them, both in wild as well as in gene bank. So this is a present status of the plant diversity of Andaman Nicobar Islands on the behalf of Botanical Survey of India and my own behalf. I would like to say, save the forest, save our life. Thank you so much.
Thank you, sir. Thank you, sir. I am requesting participants, if you have any doubts, please can interact with sir. I hope no one has any doubts, sir.
Shall I ask, sir?
Yes.
Sir, can I, sir?
Please,
It's
please.
Dr. Dhanavendan from Central Silk Board, Tamil Nadu.
Yes.
Sir, we have a plan for the Andaman Nicobar Island that survey and exploration of Morus species.
Okay.
That is mulberry, sir.
Yeah.
Which time is or which season is better, because we don't have any knowledge about that, because we are joined recently. It means that is I have 7-year experience on other place, but
Yeah.
Andaman Nicobar I am not.
May to December is very favorable for the exploration.
After May month, sir.
Yes, May to December, yeah.
Exploration for specifically for mulberry, sir, because that is our target crop.
Yeah.
Yes.
Actually, here is from you belong to the Tamil Nadu, so this is also a tropical rainforest, na?
Sir.
So that time is very good in most of the.
Weather is hazy, sunshine, 63.3 degrees Celsius in Tamilian.
Yes, please.
Hello.
Yes, बोलिए. Please tell. Yes.
And you plan to in advance before 3 or 4 months and send to the letter to the PC, the forest department and tribal departments for the getting permission.
Sir.
And any kinds of help, then we will ready, no problem. You
Okay,
just
sir. Okay, thank you.
contact us.
Okay, thank you, sir.
Any other participants? Let's the time came to thanks to our speaker. Sir, it is really highly informative and a very nice presentation. And sir, thank you very much for your valuable presentation. Shows the diversity of Andaman flora. And sir, thank you very much, sir. Thank you.
Thank you so much.
Thank you.
Thank you.
And now session is closing, and next session will start on Monday. And Monday we have a good lectures are there. And first class is Gene Bank Management by Dr. Anjali Kak, head of the department, Germplasm Conservation. And another one class is Genetic Resources of Vegetable Crops. And another interesting topic is Transitive Conservation of PGR. It is in between the in situ and on-farm conservation. Transitive conservation is a new type, it's a new concept actually. Nowadays, it is people are encouraging towards the transitive conservation, and don't that classes. It is very important. And the afternoon session practicals also there. That is the procedures for long-term storage crops of orthodox seeds. I hope these conservation classes always mostly it is important for your research work. And we will meet on Monday. Today our session will be closed, and thank you one and all, and for participants and other speakers. Thank you. Thank you very much.
Sir.
Yes.
सभी अपना वीडियो ऑन कर लें, फोटो खींच लें.
Yes. I am requesting participant, please kindly switch on your videos.
Please.
We will take one screenshot, please.
प्लीज सबने ऑन नहीं किया, प्लीज सभी अपना वीडियो ऑन कर लें, प्लीज.
Dear participants, please switch on your videos. Actually, we will take one screenshot now. Last.
ठीक है सर, थैंक यू. Thank you very much.
Thank you dear. Thank you all. Thank you very much.
Thanks to.
Thank you, sir.
सर क्लोज कर दूं मीटिंग?
मिस क्लोज कर दीजिए. Thank you, Vijay Mandal ji. Thank you.
Thank you.